Hello there, and welcome to Life Beyond Seen, where curiosity gets comfortable, science speaks softly, and the stranger corners of the natural world are allowed to unfold at an unhurried pace. Tonight, we are drifting towards something very small, very real, and just unsettling enough to make you look twice at a clear stretch of water and wonder what else is moving there besides light. Our quiet subject for this evening is the eye fluke, a tiny parasitic flatworm from a group of trematodes that can spend part of its life in the eyes of fish, often in the lens or nearby tissues, where it can cloud vision and alter the way its host moves through the world. The most studied examples belong to diplostomid eye flukes such as diplostomum, parasites with elaborate life cycles involving snails, fish, and fish-eating birds. Now, I know that is not the most relaxing phrase in the language. Eye fluke. It sounds like something invented by a Victorian naturalist who wanted to make sure no one ever enjoyed a pond again. It has the bluntness of a label that skipped the poetry and went straight for accuracy. A fluke. In an eye. No need for embellishment. Nature had already done enough. And yet, as odd and uneasy as the topic may sound, it opens the door to a story that is not merely gruesome, but deeply revealing. Because this is not a tale about a monster lurking in the dark in any dramatic sense. It is about how a creature with no grand voice, no visible menace, and no special reputation outside parasitology can shape behaviour, blur vision, redirect survival, and quietly connect birds overhead, snails below, and fish in the middle as part of a cycle that has been repeating itself long before any of us were here to be disturbed by the details. Before we go any further though, let me ask you something gentle and fitting for a night like this. Where are you listening from right now? And what kind of water do you picture when you hear a subject like this? A dark lake at dusk, a reed-lined marsh, a still canal, a cold northern bay, perhaps an estuary where river water and seawater share the same quiet argument. And what time is it in your little corner of the world as you hear this? There is something oddly appropriate about speaking of hidden life when most of the visible world has softened for the evening. If this kind of slow, curious journey is your sort of winding down, do consider giving the video a like and perhaps subscribing as well. It helps this calm little space find the people who enjoy learning about strange things in a quieter way. And if you end up drifting off somewhere between snail, fish and bird, that would be entirely in keeping with the spirit of the evening. Because the truth is, the eye fluke story begins not with horror, but with scale. So many of the living processes around us are too small, too gradual, or too ordinary looking to trigger much notice at all. A fish glides through water, a snail clings to a plant stem, a gull lifts off from the shoreline. Nothing about that scene announces that a parasite may be passing invisibly between all three, threading its way through bodies and habitats with a level of timing that feels almost deliberate, even though it is the product of evolution rather than planning. Scientists studying fish disease and aquatic ecosystems describe these trematodes as having a complex three-host life cycle. In broad terms, adult flukes live in the intestines of birds. Their eggs are released into the water, early stages infect snails, and the snails then shed large numbers of free-swimming larvae known as cercariae that go on to infect fish. In the fish, the parasite reaches the eye or nearby eye tissues and develops further, waiting for the fish to be eaten so the cycle can continue in a bird. It is efficient in the cold way nature often is, and strangely elegant in the same breath. That is one of the reasons these parasites hold the attention of researchers. They are not simply odd, they are informative. They tell us something about ecosystems as a whole, because they require several organisms to be present in the right places at the right times. They tell us something about anatomy, because some species are so specialized that they target the lens, the retina, or other ocular tissues with remarkable consistency. They tell us something about damage that can be subtle at first and serious later, because fish infected by eye flukes may develop cataracts, retinal injury, impaired vision, altered feeding, poorer movement, and increased vulnerability to predators, The eye, after all, is not a decorative detail in a fish. It is a survival tool. A fish uses vision to find food, detect danger, orient in a crowded environment, and respond quickly when the shape above the water is not a cloud but a beak. If that window begins to cloud, even slightly, the entire rhythm of life can change. And there is something especially unnerving about a parasite choosing sight. A parasite in the gut feels, in some strange way, expected. A parasite on the skin sounds unpleasant but familiar. But the eye carries a different emotional weight. We protect it without thinking. We flinch when anything comes near it. Even hearing about dust under an eyelid is enough to make most people blink in sympathy. So when we learn that a flatworm larva can take up residence in the eye of a fish, the reaction is immediate and very human. We lean in, then we recoil. We are fascinated, then mildly offended on behalf of the fish. It feels like a boundary has been crossed. That reaction is part of what makes this topic so powerful for slow science storytelling. It is not merely obscure. It sits at that exact point where biology becomes personal, not because the parasite is after us in any ordinary sense, but because we understand instinctively that vision matters. We know what it means to rely on clear sight without noticing that we rely on it at all, and the moment it is threatened, everything else comes into focus. Rare human ocular trematode infections do occur, but the cases described in medical literature are unusual and accidental. The familiar fish-eye flukes are chiefly part of aquatic wildlife cycles, involving snails, fish, and birds rather than routine human infection. What makes the subject even more curious is that the parasite does not need to destroy everything to succeed. It does not have to announce itself dramatically. In many cases, it only needs to change the host enough blur to affect prey capture, enough damage to slow reaction time, enough lens opacity to make contrast harder to read, enough disorientation for a fish to become just a little easier to catch than the one swimming beside it. Studies on eye flukes in fish have linked infection burden with cataract intensity, and researchers have also described retinal damage and changes consistent with reduced visual performance. Other work suggests that the effects of infection can ripple outward into feeding behaviour and predator avoidance, which is a very efficient arrangement from the parasite's point of view, because the bird that catches the impaired fish becomes the place where the adult fluke reproduces. It is a cycle that makes more sense the longer you stare at it, though it does not become more comforting. If anything, it becomes more impressive in a way that leaves you wondering how many invisible bargains structure the natural world while we go about calling some waters peaceful. There is also a useful correction tucked quietly inside the title of tonight's subject. When people hear a phrase like, the ocean parasite that lives inside eyes, the imagination naturally reaches for deep salt water, strange pelagic fish, and something with an almost mythical marine menace. But many of the best-known eye flukes, especially the Diplostomum species that dominate much of the research literature, are most often associated with freshwater and brackish systems, including lakes, rivers, marshes, reservoirs, and coastal zones where fish, snails, and birds overlap. They are not only creatures of one dramatic setting, they belong to connected waters, to ecologies shaped by shorelines, shallows, runoff, season, temperature, and the ordinary persistence of hosts sharing space. That matters, because it places the story closer to human life than a purely deep ocean tale would. These parasites inhabit waters people fish, farm, study, boat across, and in some cases bathe in. Scientists monitoring lakes and fish populations pay attention to them, not just because they are weird, but because they may influence fish, health, survival, food webs, and local ecological balance. What I find especially interesting is that this is one of those subjects where disgust and respect arrive together. The first response is obvious enough. No one hears larval flatworm in the eye and thinks, how charming. But once the initial shiver passes, there is another reaction waiting behind it. Curiosity. How does it get there? Why the eye? How does a snail become part of this? Why would a bird's breakfast matter to a fish's eyesight? How can something so small have consequences large enough to show up in behaviour, pathology, and population studies? These are the questions that make parasitology so strangely compelling. Parasites are easy to dismiss as freeloaders with bad manners, but they are also among the clearest examples of evolution solving problems without caring whether the solutions feel acceptable to us. They are specialists in access, timing, persistence, and transition. They do not build tools, write laws, or invent machines, yet they can navigate multiple hosts, survive hostile environments, exploit weak points, and reproduce by folding themselves into the habits of other species. That is a severe kind of intelligence, not in the conscious sense, but in the sense that selection has sanded away the unnecessary until only effectiveness remains. Eye flukes are a very neat example of that. Awful, efficient, hard to ignore once noticed. Even the name of one of the fish diseases tied to these parasites has a clipped matter-of-fact quality to it: diplostomiasis. It sounds almost bureaucratic, as though one might receive it in a filing cabinet rather than a body of water. Yet behind that tidy label is a process that can include penetration through fish tissues by cercariae, migration toward the eye, development into metacercariae, and varying degrees of ocular harm depending on parasite load, host species, and parasite species. Some reports describe heavy infections leading to blindness or mortality, while others emphasize sublethal effects that still matter because they alter how fish feed, evade, and survive. Researchers have documented these infections in wild populations and in aquaculture contexts as well, which tells us that eye flukes are not just a curiosity of untouched places. They can become relevant anywhere the ecological pieces line up. And because the life cycle depends on more than one host, changes in habitat, water conditions, snail abundance, and bird use of an area may all influence transmission in ways that are scientifically important, and, for managers of fisheries and vulnerable fish populations, practically important too. Still, for all the science, there remains something almost intimate about the subject. A fish does not know it is serving as the middle passage in a trematode life cycle. It only knows light, motion, contrast, hunger, current. It knows the dart of prey and the shadow of threat. If a lens begins to cloud, there is no diagnosis. There is only a world that becomes harder to read. That, to me, is where this story becomes more than a medical oddity. It becomes a quiet reminder that survival is often negotiated through tiny anatomical advantages we barely think about until something interferes with them. Clear sight is one, quick detection is another. The ability to judge direction in a split second may be the difference between feeding and fleeing, between living another day and becoming the next step in a parasite's itinerary. When researchers report impaired retinal function, lens damage, cataract formation, or fish becoming easier prey for birds, those findings are not abstract. They describe a narrowing margin of safety inside an already difficult world. So as we ease into this topic properly, there is no need to rush toward the most dramatic image and stay there. The eye fluke story is stronger when taken slowly. A bird in the shallows, eggs entering water, a snail acting as the first workshop for multiplication, larvae released in astonishing numbers, a passing fish, a successful penetration, a journey inward, an eye becoming not just an organ of sight but a temporary address. That is the strange machinery we're about to follow. Not because it is merely grotesque, though it certainly qualifies from time to time, but because it reveals how much of nature runs on relationships too small for casual notice and too consequential to dismiss. And the closer one looks, the clearer it becomes that this is not simply a story about something living inside an eye. It is a story about movement between hosts, about biology taking the shortest available route to continuation, and about how an entire chain of life can hide inside a detail no larger than a fish's gaze. That is where we can begin to follow the parasite itself. Not at the moment it startles us, but at the moment its long journey first becomes possible. If we stay with the parasite itself for a while and follow it in the plainest possible way, what becomes striking is not just where it ends up, but how methodically it gets there. An eye fluke does not begin life in an eye. It does not even begin life in a fish. The well-studied diplostomid eye flukes, including species of Diplostomum, usually follow a three-host life cycle that moves through a bird, a snail, and then a fish before returning to a bird again. Adult worms live in the intestines of fish-eating birds. Eggs leave the bird in its droppings and enter the water. From there, the parasite's early stages must reach a suitable snail, where asexual multiplication produces large numbers of mobile larvae. Those larvae leave the snail, find fish, penetrate them, and migrate to the eye or nearby tissues, where they develop into metasacchariae. The cycle is only completed when an infected fish is eaten by a fish-eating bird. That is the broad outline, and even in its simplest form it already sounds like a route planned by someone with an unreasonable amount of patience. It is not planned, of course, it is selected. Generation after generation, anything that failed to move successfully from host to host was lost, and what remains is a life cycle that looks almost deliberate because inefficiency has been trimmed away for a very long time. There is also a useful correction to keep gently in mind as we move through this. The title may suggest an ocean parasite in the strictly marine sense, but many of the classic eye flukes studied in fish, especially Diplostomum species, are common in freshwater and brackish environments, where snails, fish, and fish-eating birds overlap. Lakes, rivers, marshes, reservoirs, nearshore shallows and mixed waters can all provide the right arrangement. In other words, this is not a creature of one dramatic abyss. It belongs to linked habitats, to bird roosts and snail-rich margins, to nursery waters where young fish feed, and where a parasite has far more opportunity than it would in some clean and empty theoretical pool. Scientists studying these trematodes describe them as widespread fish parasites with complex life cycles, and that complexity matters because every stage depends on the right host appearing at the right point in the sequence. A parasite like this does not survive by brute force. It survives by getting the timing right often enough. That is a quieter talent, but in evolutionary terms it is probably the more dangerous one. The first stage of the story takes place in a bird, usually a fish-eating bird that serves as the definitive host. That term sounds rather grand, but it simply means the host in which the parasite reaches adulthood and reproduces sexually. Once there, adult flukes live in the intestine and produce eggs that pass out with the bird's feces into the water below. It is a thoroughly unsentimental arrangement. The bird flies, feeds, roosts, and goes about being a bird, while the parasite uses those habits as transportation and distribution. A gull, a tern, a grebe, another fish-eating water bird. From the parasite's point of view, these are less characters than destinations. What matters is not personality, but plumbing. The adult stage must be in a place where eggs can exit and reach water, because without water, the next stage never begins. That dependence already tells you something important about the parasite's life. It is not free in the way we tend to imagine living things being free. It is more like a specialist courier with a route fixed by anatomy, habitat, and chance encounters. Every success depends on the host doing normal host things. The bird does not cooperate out of kindness. It merely continues living, and that is enough. Once the eggs are in water, the parasite shifts into one of the recurring themes of trematode biology, which is that each stage is built for a particular job and very little else. In Digenean trematodes, the life cycle commonly includes an egg, a miracidium, stages within the snail such as sporocysts and often rediae, then a cacaria, then a metacacaria, and finally the adult worm. That list is not just terminology meant to burden biology students. It is a map of changing bodies for changing tasks. The miracidium is the stage that emerges from the egg and infects the first intermediate host, usually a snail. Inside that snail, the The parasite multiplies asexually, producing later larval generations that eventually give rise to cercariae, the mobile infective larvae released back into the water. This multiplication step is one of the great practical tricks in trematode life cycles. A single successful entry into a snail does not merely preserve one parasite. It can become a production site, a living factory that turns one successful beginning into many thousands of chances later on. To put it less charitably, the snail is where the parasite stops being a traveller and starts being an enterprise. And that first intermediate host, the snail, is not a minor detail tucked between the more dramatic moments. It is the engine room. In diplostomid eye flukes, freshwater snails are central to transmission, and limnid snails are among the hosts repeatedly noted for diplostomum. Inside the snail, the parasite does not behave like a quiet tenant. It reproduces asexually and generates the free-swimming forms that will later leave in large numbers. USGS researchers working on eye flukes in Upper Klamath Lake noted that snails are capable of shedding tens of thousands of infective parasites into the water every day, which gives a sense of the scale involved. When people hear tiny parasite, they often imagine rarity, but small does not mean scarce. A life cycle like this succeeds partly because the snail stage can flood the water with opportunity. Most of those larvae will fail. They will miss fish, die, disperse badly, or simply run out of time. The parasite solves that problem the way many organisms solve it. Not by guaranteeing individual success, but by producing enough attempts that some succeed anyway. It is quantity standing in for certainty. Nature does this constantly. We only call it sinister when the details happen to involve eyes. What emerges from the snail is the cercaria, and this is where the life cycle becomes much easier to picture. Cercariae are motile larvae, built to leave one host and locate another. General trematode references describe the cercaria as a stage specialized for locomotion, host finding, invasion, and then transformation once entry is achieved. They do not feed in the way a leisurely creature might. They are better understood as short-lived delivery systems with one assignment: get out. Find the next host. Get in. In eye flukes, these cercariae are released into the water column, where they swim and search for fish. It is an intensely exposed part of the life cycle. The parasite is no longer protected by intestine or snail tissue. It is out in open water, vulnerable to currents, temperature, light conditions, timing, and sheer bad luck. Yet this stage is effective enough that fish in suitable habitats can be infected in substantial numbers. There is something almost bluntly efficient in the design. No wasted personality, no decorative flourish, just movement, contact, penetration, transformation. If a human project were built with that level of singular focus, it would be admired in a business magazine and feared by everyone else. Finding a fish is only the next problem, not the final one. Once a cercaria encounters a suitable fish, it penetrates the skin or gills and begins to migrate. That detail matters because it shows that the eye is not the entry point in the ordinary sense. The parasite does not need the fish to look directly at it in a particularly unfortunate moment. It gains entry through body surfaces and then travels internally to its preferred site. For diplostomum eye flukes, that preferred site is often the eye lens, though other diplostomids may occupy different liquid regions or structures of the eye. Researchers describe this migration to the lens as central to the parasite's strategy. Once there, the cercaria transforms into a metacercaria, a larval stage adapted for waiting in the fish until the fish is eaten by a bird. One study summary notes that the parasite quickly invades the fish's eye lens, where it is protected from the host immune system. That is a remarkable sentence when you stop and sit with it for a moment. Protected from the immune system. The lens is not just a location with useful consequences for vision, it is also a shelter. The parasite is not merely harming sight, it is choosing a place where the host has limited ability to fight back. That is not personal malice, It is simply very good parasite design which is somehow worse. From the fish's side, this stage can look like bad luck arriving from nowhere. A young fish in snail-rich water is feeding, turning, schooling, avoiding shadows, and doing all the ordinary things required to remain alive. Meanwhile, Cercaria in that same water are attempting to invade. Laboratory and field work on eye flukes has shown that the consequences depend on infection intensity, host species and parasite species, but the logic stays steady. If the parasite reaches the eye and develops there, it may induce cataracts, damage retinal tissues, reduce visual performance, and alter behaviour in ways that can make the fish easier for birds to catch. That is why the life cycle is so tightly organised around vision. The fish is not just a container, it is a transport host that must remain alive long enough to be eaten by the correct predator. If the parasite kills too early, it loses. If it does too little, the fish may survive and escape predation for too long. So what we often see are effects that interfere enough to matter. Reduced shoaling, weaker escape responses, more time near the surface in some systems, poorer feeding or avoidance. Not always the same pattern in every fish, but the general direction is clear. A fish that cannot read its world properly becomes easier to remove from it. That intermediate period inside the fish, when the parasite is a metacercaria, is also a lesson in patience. The parasite has arrived at one of its most advantageous addresses, but it still has not reached adulthood. It cannot reproduce sexually there. It has to wait until the fish is eaten by the right sort of bird. This is one of the more interesting things about parasitic life cycles. They're often built on delayed success. Parasite must be content, if that word can be borrowed loosely, to occupy an unfinished stage for as long as necessary. And because the metacercaria is positioned in such a consequential organ, the waiting is not passive in effect, even if it is passive in appearance. Infections in the eye lens can create cataracts by disrupting lens structure, and eye flukes in other parts of the eye may affect visual processing in different ways. Behavioural studies cited in review material describe infected fish as more vulnerable to simulated bird predation, less cohesive in shoals, and less effective in anti-predator responses. All of that serves one endpoint. A fish-eating bird must catch and swallow the fish. Only then does the parasite move from waiting room to destination. When that bird finally eats the fish, the cycle closes in a way that feels both tidy and slightly rude. The metacercaria, which spent part of its existence compromising the fish's sight, now reaches the host, where it can mature into an adult fluke in the intestine. There it reproduces, sheds eggs into the environment through the bird's feces, and starts the whole process again. This circular structure is one reason parasitologists are so interested in host connections rather than isolated infections. An eye fluke is not fully understandable if you look only at the fish. You have to include the birds overhead, the snails below, the seasonality of shedding, the physical character of the water, and the density of hosts. Even environmental changes can matter. USGS work in Upper Klamath Lake has linked concern about eye flukes with warming temperatures, habitat alteration, nutrient inputs, snail abundance, and juvenile fish mortality, because more snails can potentially mean more parasites in circulation. That does not mean every warm pond becomes a horror film. It means transmission is ecological. Parasites are woven into the same conditions that shape everything else in aquatic systems. When water, habitat, and host communities shift, parasite opportunity often shifts with them. Another thing worth noticing is that this route through life is both highly specialised and surprisingly flexible. Review and molecular work on Diplostomum shows a broad geographic distribution, and researchers continue to refine species identities because morphology alone has often been insufficient. Some species have different preferred fish hosts. Some target different eye structures. Some may show different degrees of host specificity or regional variation. But the general plan remains recognisable. Bird, water, snail, water again, fish, then bird again. It is a route based on recurring contact between habitats and behaviours that have existed for a very long time. Birds feeding on fish, snails grazing in shallow water, juvenile fish moving through zones where larval parasites can find them. That is why parasites like this can persist so successfully. They do not need modern circumstances to exist. They only need host relationships that keep happening, and in aquatic ecosystems those relationships are ancient. From the parasite's view, a shoreline crowded with life is not scenery, it is infrastructure. There is something else that becomes clearer when you follow the whole route carefully. The eye fluke does not decide on the eye because the eye is symbolically dramatic. It gets there because the eye offers a combination of practical advantages: a predictable target in a fish host, a site where damage can alter behaviour and vulnerability, and in some cases a location such as the lens where immune access is limited. That means the parasite's journey through life is not random wandering that happens to end somewhere memorable. It is a chain of selected solutions: egg in water, larva to snail, multiplication, Cercaria out into the water, penetration of fish, migration to the eye, waiting in the fish, bird predation, adult reproduction. If it sounds repetitive, that is because it is. Repetition is the point. Most of life at scale is repetition with slight variation. We notice the spectacular event, but evolution works by keeping the route that works often enough. Eye flukes are unsettling precisely because their route is so plain once you understand it. There is no mystery machine hidden behind the curtain. Just a series of small successes, each one dependent on anatomy and timing, each one enough to carry the parasite forward. So when we say that a tiny parasite finds its way through life, what we really mean is that it passes through several lives by being extremely good at each brief stage it inhabits. It begins in one body and exits into water. It enters a snail and multiplies. It leaves again as something built for movement. It penetrates a fish and settles where sight can be bent to the parasite's advantage. Then it waits for a bird to make the final connection. It is not elegant in a sentimental way, and there's nothing comforting about it if you happen to be a fish. But it is coherent, and in biology, coherence is usually the closest thing to genius that nature allows. Once you see the whole route, the eye itself stops being a random place where something horrible happened and becomes part of a larger, colder logic. And that larger logic becomes even clearer once we slow down and look at the three-way passage in order. Because the next part of the story is not simply that the parasite lives in several hosts, but that each host gives a different kind of life. The route from snail to fish to bird only sounds strange until you look at how neatly each host solves a different problem for the parasite. The snail is where the numbers are made. The fish is where the parasite settles into a form that can wait and interfere with sight. The bird is where adulthood begins and reproduction finally happens. In the best-studied eye flukes, including species of Diplostomum and related diplostomids, that sequence is not optional decoration around the edges of the story, it is the story. The life cycle depends on a piscivorous bird as the definitive host, a mollusk, usually a freshwater snail, as the first intermediate host, and a fish as the second intermediate host. Only when all three are available in the same connected habitat can the cycle keep turning. The bird comes first in the biological sense, even if it is often the last thing people picture. Adult flukes live in the intestine of fish-eating birds and produce eggs there. Those eggs leave with the bird's feces and drop into water, which is both undignified and highly effective. Parasites are rarely interested in respectable exits. They're interested in reliable ones. A gull feeding over a lake, a grebe diving, a heron taking fish from a quiet margin. These are not just scenes from a wetland. They are transport links in a trematode network. Studies on Diplostomum have specifically documented adult worms in the intestines of gulls, including herring gulls and common gulls, and the broader life cycle literature consistently places the adult stage in piscivorous birds. Once the eggs enter the water, the parasite changes from something contained inside a bird into something exposed to the outside world. Trematodes do not remain in one form for long if the next task requires a different design. A general trematode life cycle includes an egg, then a miracidium that infects a snail, then later larval stages within the snail, and eventually a cercaria that leaves the snail to seek another host. It sounds like a committee was allowed to name things in sequence, but the biology behind those labels is extremely practical. Each stage is built for one assignment, and then replaced by another stage better suited to the next step. That is one reason parasites like these can seem so efficient. They are not trying to make one body do everything. They keep changing the body to match the job. The snail is where the pace of the story changes. Up to this point, the parasite has mainly been moving from place to place. Inside the snail, it begins to multiply. This is the first major advantage the life cycle gains, and it is a substantial one. A single successful parasite entering a suitable snail can produce large numbers of offspring through asexual reproduction. USGS researchers studying eye flukes in Upper Klamath Lake note that infected snails can shed tens of thousands of infective parasites into the water every day. That figure is worth sitting with for a moment, because it explains how a tiny parasite compensates for the fact that open water is a difficult place in which to find one specific fish. The parasite does not solve the problem by becoming a better navigator in some grand individual sense. It solves it by creating an enormous number of attempts. If enough cercaria leave the snail, some will encounter fish even when most do not. That makes the snail less like a minor stop, and more like the manufacturing district of the entire system. It is where the parasite moves from scarcity to surplus. A fish may only ever carry a limited number of parasites in its eyes, and a bird may only host a certain number of adults in its intestine. But a productive snail can release wave after wave of cercariae into the surrounding water. That is why parasitologists and fish health researchers often pay close attention to snail abundance, snail species, and the seasonal timing of snail shedding. If you want to understand why fish are being exposed at a particular place and time, the most useful question is often not which fish are sick yet, but which snails are already acting as launch points. In Upper Klamath Lake, for example, scientists have been examining whether altered environmental conditions and increased snail abundance may help explain greater fluke pressure on juvenile fish. Then, the cercariae leave. This is one of the most exposed parts of the parasite's life. Inside a bird, the adult is housed and fed. Inside a snail, the larval stages are protected while they multiply. Once the cercaria enters the water, it has very little room for error. It must swim, encounter a suitable fish, penetrate, and begin internal migration before its limited time runs out. Laboratory work on Diplostomum spathaceum cercario showed that survival and infectivity are strongly time-dependent, with mortality rising sharply after about 20 hours at a constant temperature in the experimental setting. In other words, these larvae are not drifting around for days enjoying the scenery. They're operating on a short schedule. That lends the whole stage a feeling of urgency, not panic exactly. Parasites do not panic, but certainly no dawdling. The next host, the fish, is not infected through the eye itself. That detail often surprises people, perhaps because the title of the subject encourages the imagination to leap straight towards something entering the pupil, like a villain in a low-budget underwater thriller. The actual route is less theatrical and more efficient. Cercariae encounter the fish and penetrate through the skin or gills. From there they migrate through the host toward the eye, often reaching the lens with striking speed. One immunology paper on Diplostomum pseudospathaceum notes that the parasite quickly invades the fish's eye lens, where it is protected from the immune system. Other summaries describe the same sequence plainly: skin penetration, migration to the eye lens, then transformation into the metacercarial stage. So the eye is not the doorway, it is the destination. That destination is not chosen at random. The lens gives the parasite two important advantages at once. First, it is a place where even modest damage can matter a great deal to the fish. Second, it is a site where the parasite is relatively protected from immune attack. That combination is about as good as a parasite could ask for. It can survive there while also affecting the host in ways that may benefit later transmission. The literature on eye flukes repeatedly connects lens infections with cataract formation, impaired vision, and in heavier cases blindness or marked reductions in normal performance. Retinal damage has been described as well, depending on the parasite species and site of infection. For a fish, that is not a minor inconvenience. Fish use vision for feeding, orienting, maintaining distance within shoals, and detecting predators above and around them. If that window clouds, everyday survival becomes more difficult almost immediately. What happens next is one of the colder parts of the story, though it is also one of the most revealing. Once in the fish's eye, the parasite does not need to race onward. It can wait as a metacercaria. This waiting stage is not biologically lazy. It is strategic. The fish is not the final home. The fish is the bridge to the final home. The metacercaria remains in the fish until the fish is eaten by a bird, and only then can the parasite mature into an adult and reproduce sexually. That means the parasite benefits if the fish becomes easier for the right predator to catch. USGS descriptions of the Diplostomum gavium cycle state the point directly. Infected fish develop problems such as impaired vision and become easier prey for water birds, allowing the parasite to complete its life cycle by moving from fish back to birds. There is good evidence that this increased vulnerability is not just a convenient idea on paper. Experimental work has shown that eye fluke infections can weaken anti-predator responses and raise susceptibility to bird predation. One study found that stronger parasite-induced cataracts were associated with weaker responses to simulated avian attack. Another field-linked study examined whether infected fish were more likely to be taken by bird-definitive hosts, and found support for the idea that eye flukes predispose fish to predation, thereby enhancing transmission. That does not mean every infected fish swims upward and volunteers to be lunch. Nature usually works by shifting odds, not by writing dramatic final speeches. But even a modest increase in predation risk can matter enormously from the parasite's perspective if it helps enough metacercari reach birds. A detail that makes this journey especially interesting is that the parasite is relying on ordinary behaviour at every step. The bird does not have to do anything unusual. It eats fish and defecates into or near water. The snail does not have to become a tragic figure in a biology lesson. It simply serves as the first intermediate host while the parasite multiplies within it. The fish does not need to make a poor life decision. It only needs to inhabit water where cercariae are present. The parasite thrives by slipping into predictable interactions that were already going to happen. This is one of the reasons trematode life cycles can be so stable over long periods. They do not depend on rare events. They depend on ordinary ecology repeated often enough. A shoreline full of snails, juvenile fish moving through shallow water, and birds feeding overhead is not unusual. For the parasite, it is ideal, and still, even with all that repetition, success is never guaranteed. One laboratory study on Arctic char reported that of the Diplostomum cercariae presented to fish, about 19% penetrated, and of those that penetrated, 46% migrated successfully to the lens. Those numbers are useful because they show two things at once. The parasite is effective, but it is not magical. Many cercariae fail. They miss, they die, or they enter without completing the journey. This is exactly why the snail stage matters so much. The system is built around a lot of attrition. If the parasite was stingy with larval output, the whole arrangement would collapse under the weight of its own inefficiency. Instead, it sheds large numbers and accepts that most will not make it. This is not elegant in the sentimental sense, but it is mathematically persuasive. There is also variation hidden inside what sounds like one clean route. Different diplostomid species can target different structures in the eye, and not all eye flukes behave in exactly the same way. Some occupy the lens, some the vitreous body, and related diplostomids have different host associations and tissue preferences. Taxonomic work has shown that the group is more diverse than older morphology-based identifications often suggested. That matters because when people hear "eye fluke," they may imagine one standardized creature doing one standardized thing everywhere. The real picture is more crowded. There are multiple species, multiple host combinations, and regional differences in which snails, fish, and birds are involved. But the broad route remains familiar enough that once you learn the pattern, you begin to recognize variations on it. The details change. The arrangement does not. Another practical point in the snail to fish to bird journey is that timing matters as much as anatomy. Cercarial release from snails can vary seasonally, and fish exposure can rise or fall with temperature, habitat conditions, and the abundance of infected snails. Researchers in Klamath have specifically emphasised the need to understand how eye fluke communities change seasonally, because that timing may help explain when juvenile fish face the greatest risk. This is worth noting because parasite stories are often told as though infection is a constant background menace, equally present at all times. In reality, transmission often comes in pulses. A young fish passing through one bay in early summer may be facing a different level of exposure than another fish in cooler conditions or in a habitat with fewer infected snails. The life cycle is stable, but the pressure it creates can still rise and fall with the calendar and the weather. The bird stage, when it comes again at the end of this route, is almost blunt in its simplicity. The infected fish is eaten. The metacercaria reaches the bird's gut. It matures into an adult worm. Reproduction begins again. Eggs return to water. If you are the parasite, this is success. If you are the fish, it is not the sort of career progression one hopes for. Yet biologically, it is a very direct example of trophic transmission, where moving up the food chain is not a side effect, but the central requirement. The parasite uses predation as transportation. That is one reason eye flukes have been such useful systems for studying host manipulation, cataracts, predator-prey effects, and the ecological roles of parasites in food webs. They do not merely infect one organism in isolation. They connect the behaviour and survival of different species across the same habitat. By the time you trace the whole journey carefully, the parasite starts to look less like a random threat and more like a traveller using a very old route. Not a traveller with luggage and a charming destination photo, of course. More like one of those relentless commuters who always knows which platform to stand on and somehow never misses the train. Snail, fish, bird. Each host provides something the next one cannot. The snail provides multiplication. The fish provides protection, growth, and a chance to alter predation risk. The bird provides adulthood and egg production. Remove any one of those stations and the route breaks. Leave them all in place, and the parasite can keep cycling through the system year after year with a persistence that is easy to underestimate until you realise how little fanfare it needs. And once that route is clear, the next part becomes easier to understand. Because what really gives the eye fluke story its strange force is not just that it moves between hosts, but that it sometimes reaches the eye with enough precision and enough consequence to turn a fish's vision into the most useful stop on the entire journey. The phrase "wrong turn" makes immediate sense to us because the eye feels like one of those places where nothing extra should ever arrive. A speck of dust feels intrusive there. A stray eyelash can derail an entire afternoon. So when a parasite enters the picture, the mind naturally treats the event like a navigation error, as though some tiny creature was meant for an intestine, missed an exit, and ended up in the worst possible room in the building. For many fish eye flukes, though, that is not what is happening at all. In the diplostomid species most often discussed in fish, including Diplostomum pseudospathicium and related eye flukes, the eye is not an accident. It is the target. Cercariae released from infected snails penetrate the fish through the skin, then migrate toward the head and reach the eye lens quickly, often within 24 hours, where the parasite is relatively sheltered because the lens is an immunologically privileged site. From our point of view, it looks like a wrong turn. From the parasite's point of view, it is a very successful arrival. That correction matters because it changes the mood of the whole story. A random mistake is unsettling in one way. A precise destination is unsettling in another, much deeper way. The fish is not simply unlucky because something wandered into its eye. The parasite has been shaped over many generations to get there. Studies of Diplostoma migration show that after penetration the larvae orient within host tissues toward veins, the head, and the eye. Experimental infection work in rainbow trout found that Metacariae could first be detected in the lens as early as 5 hours after exposure and continued to accumulate through the next several hours, with success depending partly on where the Cercaria entered the fish. Parasites entering nearer the head were more likely to reach the lens than those entering through the tail. That is not the pattern of a creature blundering around in the dark. That is the pattern of a traveller with a map, even if the map exists only as evolved responses to tissue cues, gradients, and anatomy rather than anything we would recognise as intention. The route itself is rather blunt once you picture it in slow motion. The Cercaria leaves the snail and enters open water with very little time to spare. If it contacts a fish, it penetrates through the skin or gills and begins moving inward. It does not swim through the pupil like a villain making an entrance. It comes in through the ordinary exterior of the fish, then threads its way through tissues toward the eye. A workshop summary on eye diplostomiasis in rainbow trout describes the Cercaria as penetrating the fish at any point on the surface and actively migrating to target tissues such as the lens, retina, or vitreous body, mostly through subcutaneous connective tissue and skeletal muscle. The same summary notes that the earliest detection in the lens can occur 5 hours after emergence from the snail host. One older infectivity study likewise showed that lens establishment can be rapid, and that entry closer to the head gives the parasite a much better chance of success. What sounds dramatic in the title is, biologically speaking, a very practical series of movements through a fish that never agreed to be used as internal transit space. Once the parasite reaches the eye, the choice of location begins to look grimly sensible. The lens is useful not because it is symbolically disturbing, though it certainly is, but because it solves two problems at once. It gives the parasite a place where damage can interfere with the fish's vision, and it also gives the parasite a degree of protection from the host's immune defences. Researchers describing Dius pseudospathicium note that the larvae migrate to the immunologically inert or privileged eye lens, where they grow until the fish is eaten by a bird. In other words, the lens is not merely a place to hide, it is a place to wait productively. The fish continues living, swimming, and trying to function, while the parasite occupies a site that is difficult for the immune system to clear and influential enough that even modest damage may matter. That is one of the reasons eye flukes are so effective as trophically transmitted parasites. They do not need to kill their host immediately. They only need to make survival a little harder and predation a little easier. And this is where the eye changes from a destination into a workplace. Infection of the lens is strongly associated with cataract formation, reduced visual ability, and greater vulnerability to predators. Experimental and review work on Diplostomum repeatedly links eye fluke infections with poorer escape responses and increased susceptibility to aerial predators, which matters because a fish-eating bird is the parasite's final host. That means the visual damage is not just collateral mess, it fits the larger route. A fish that sees less clearly may feed less efficiently, orient less well, respond more slowly, and spend more time in risky positions. One study reported that fish infected with D. spathicium had decreased escape responses and increased susceptibility to artificial aerial predators. Another open-access immunology paper summarizes the same pattern in plain terms. Cataracts reduce visual ability and likely predispose infected fish to predation by the final bird host. The parasite is not making the fish theatrical, it is making the fish fractionally less competent in a world where fractions matter a great deal. Still, the eye is not one perfectly uniform destination across all eye flukes, and this is where the phrase wrong turn begins to recover some truth. Some Diplostomum species and related diplostomids do not remain neatly in the lens alone. Work on wild three-spined sticklebacks found eye fluke larvae in the subretinal space, where they caused retinal detachments and damaged the retina and retinal pigment epithelium. In those fish, the eye was still the organ being targeted, but the exact tissue site mattered, and the resulting damage could be severe in ways that went beyond the classic cloudy lens picture. Other sources note that metacercaria may occur in the lens, vitreous humour, or retina depending on the species and host combination. So even within the broad category of eye flukes there is variation. The parasite may arrive at the eye on purpose, but what happens once it gets there is not always identical. Different tissues mean different consequences, and the fish pays for all of them in one currency, a world that becomes harder to read. There are also cases where the parasite seems to overshoot, misplace itself, or at least participate in pathology that looks less cleanly planned than the standard lens story. A 2025 study on bullhead catfish described an unusual localization in which Diplostomum metacercaria were displaced into a sac protruding from the lens. The authors suggested this might represent a host defense response or tolerance mechanism, rather than the parasite's preferred original niche. They found that sac formation was common in infected bullheads, especially at higher parasite intensities, and that the process appeared linked to altered pathology around the lens. That is a useful reminder that the host is not merely a stage set while the parasite performs. Hosts push back, compensate, remodel tissue, and sometimes force the parasite into odd arrangements. In such cases, wrong turn may not mean the parasite lost its way on the road to the eye. It may mean the host changed the road after the parasite arrived. The fish eye, then, is both a destination and a negotiation. Parasites get there with remarkable speed and reliability, but arrival does not guarantee a perfectly smooth occupation. Lens opacification, capsular damage, retinal effects, vitreous inflammation, even unusual sac formation in some host species, all suggest that the eye becomes a site of struggle as much as shelter. A rainbow trout workshop summary described tiny capsular perforations at the site of entry into the lens, exudation of cortical lens fibres, retinal detachment in chronic infections, and fish that become dark, unable to feed properly, more prone to secondary infections, and more susceptible to predation. These are not decorative side effects. They are the anatomy of a fish losing clarity while still being expected to function. And because fish vision is tied to feeding, schooling, predator detection, and spatial coordination, even partial impairment can ripple outward into behaviour long before total blindness is reached. From a storytelling point of view, that is why the eye fluke unnerves people more than many other parasites. It is not just that it is small and hidden, it is that its chosen location makes intuitive sense to us in the worst possible way. We know what eyesight does, so we immediately understand what it means to compromise it. We do not need a lecture in fish neurobiology to grasp the consequence of a lens turning white or a retina detaching. The science simply sharpens what instinct already tells us. Experimental work has shown reduced vigilance, cataracts, weaker escape reactions, and increased vulnerability to predation in infected fish. The eye is where perception becomes behaviour, and behaviour is where survival begins to slip. That is why a parasite in the eye feels more personal, more precise, and somehow more impolite than one hidden somewhere deeper and less obvious. It is not merely occupying tissue, it is interfering with the host's way of knowing the world. There is, however, another sense in which a parasite can truly take a wrong turn into the eye, and that is when it ends up in an accidental or aberrant host. This is uncommon for the fisheye flukes that dominate the story, but trematodes broadly do sometimes appear in human eyes or other unusual hosts. The clearest modern examples come from avian eye flukes of the genus Philophthalmus, which normally parasitize the conjunctival or orbital tissues of waterbirds. Human infection is rare, but it has been documented. A 2019 case report described a Philophthalmus trematode extracted from the bulbar conjunctiva of a patient in southern Texas, and the authors noted that human philophthalmiasis is rare, with only about 24 reported cases in the literature at that time. A 2025 CDC report described Philophthalmus lacrymeus in South America and explained that these avian eye flukes usually inhabit the conjunctival sac of water birds, with freshwater or marine snails as intermediate hosts and water birds as final hosts. In birds, the route can involve migration through the lacrimal ducts to the orbital cavity after excistation. In a human, though, the parasite has arrived somewhere that is biologically possible but evolutionarily off-script. That is a real wrong turn in the stricter sense. The right organ, perhaps, but the wrong species. That distinction is important because it keeps the biology honest. In fish infected by classic diplostomid eye flukes, the parasite in the eye is not an accident. In a rare human conjunctival infection by an avian eye fluke, the eye may represent a spillover event into an aberrant host. These are both eerie, but they are eerie in different ways. One shows the precision of a mature parasite life cycle. The other shows the looseness that can happen when ecological contact allows a parasite to end up somewhere. Evolution did not primarily prepare it to thrive. Parasitology is full of those two moods. There is the mood of grim efficiency, where every host and tissue seems selected with upsetting care. Then there is the mood of spillover and accident, where the parasite ends up where it can, not necessarily where it should. The eye happens to be one of the few organs that can make both moods memorable at once. So the title of this part can be taken two ways, and both are useful. To us, any parasite entering an eye feels like a wrong turn, because the eye is one of those structures we imagine as protected, sealed off, almost above such indignities. To the fisheye fluke, however, the path to the eye is often the correct route, the efficient route, the one that brings shelter, leverage, and a better chance of reaching a bird through predation. Only in unusual localizations, host defenses that distort the parasite's niche, or accidental infections in aberrant hosts does the phrase become literally accurate. Most of the time, the more unsettling truth is simpler than that. The parasite did not get lost. It arrived exactly where it needed to be, and once it is there, the eye stops being just a vulnerable organ and starts looking like one of the most useful addresses the parasite could possibly have found. And once that is clear, the next step is not merely to say that it lives there, but to slow down and look at what this creature actually is, how it is built for such a life, and why something so small can organise so much damage around such a delicate place. What eye flukes really are becomes much clearer the moment we stop treating the phrase like a horror title and start treating it like a zoological description. An eye fluke is not a separate kind of creature invented especially to trouble fish in dramatic ways. It is a trematode, which means it belongs to a large class of parasitic flatworms called trematoda. Flukes are flattened, unsegmented worms, usually small, often leaf-like or ribbon-like in form, with attachment structures such as suckers and a simple digestive system. Many species are hermaphroditic, and many pass through egg, larval, and adult stages while using two or more hosts. In other words, before an eye fluke is a problem in a fish, it is already part of a very old and very successful group of parasites that have made a profession out of living inside other animals. That helps because eye fluke is really a practical label, not a perfect taxonomic box. In fish pathology and aquatic parasitology, the term often refers to diplostomid trematodes, especially species in the genus Diplostomum, whose larval stages infect the eyes of fish. Researchers describe Diplostomum as containing several common parasites of freshwater and brackish water fish, and modern work also treats fisheye flukes more broadly as part of a diverse, globally distributed diplostomid group found in both marine and freshwater fish communities, so there is not one single eye fluke behaving identically everywhere. There is a cluster of related parasites, with different species, different host combinations, and different preferred tissues, all sharing the same broad habit of using the fisheye as an important stage in life. That is why the name is useful but a little misleading at the same time. It sounds as if the parasite's whole identity begins and ends in the eye, when in fact the eye stage is only one part of a longer life cycle. The adult worm usually lives in a bird, not in a fish. The fish carries a larval stage, commonly the metacercaria. Before that, a snail carries earlier larval stages and produces the free-swimming cercaria that leave the snail and infect fish. General trematode biology follows this same broad pattern. A first free-swimming larva known as a miracidium reaches a snail, transforms and multiplies inside it, and eventually gives rise to cercariae that leave the snail to infect a vertebrate host, or a second intermediate host, depending on the species. An eye fluke, then, is best understood not as a creature with one home, but as a parasite built to live different kinds of lives in different bodies. If that sounds inconveniently complicated, it is, but only from our perspective. For the parasite, this arrangement is extremely practical. One host gives it multiplication, another gives it a place to wait and develop, and another gives it adulthood and sexual reproduction. In the best-studied fish eye flukes, including Diplostomum species, fish-eating birds serve as the definitive hosts, snails act as the first intermediate hosts, and fish become the second intermediate hosts carrying the metacercarial stage. The whole design works because each body solves a separate problem. The bird is where reproduction happens. The snail is where one invading parasite can become many. The fish is where the parasite can settle into a tissue that helps it survive until the fish is eaten. That is not a messy life. It is a highly partitioned one. Each host provides a function, and the parasite changes form to fit it. The fluke's body plan makes more sense when you think of it as equipment for attachment and survival rather than graceful movement. Britannica's description of flukes emphasises that they are flattened parasites with muscular suckers, and that many species use hooks or spines for attachment as well. Their digestive system is simple, often with a mouth leading into blind-ending intestinal branches, and usually no anus. This is not luxurious engineering, it is stripped-down engineering. A fluke does not need the body of a free-swimming predator or the sensory apparatus of an active hunter. It needs to hold on, absorb nutrients, reproduce, and move through its life cycle with minimal fuss. Even the adult form is not especially interested in elegance. It is interested in persistence. That practical simplicity is part of what makes trematodes so successful across so many vertebrate hosts. Still, the adult worm is only one version of the animal. The larval stages are where the eye fluke story becomes truly distinctive. In trematodes, the body is repeatedly rebuilt to suit the next job. The miracidium is for finding a snail. The stages inside the snail are for multiplying. The cercaria is for leaving the snail and reaching the next host. The metacercaria is for settling in and waiting. That repeated redesign is one of the fluke's main advantages. Instead of forcing one body to perform every task badly, it shifts form as the demands shift. This means an eye fluke is not really one creature in one shape from beginning to end. It is a sequence of specialized versions of the same parasite, each one temporary, each one efficient, and each one discarded once the next stage is reached. It would be exhausting if it were not so effective. Inside the snail, the parasite lives in a way that is both productive and slightly impolite. After the early larval stage reaches the snail, it transforms and multiplies asexually. General trematode references describe this as the stage at which daughter generations are produced, and cercariae eventually develop. That multiplication is the reason snails matter so much in transmission. A single successful infection of the right snail does not just preserve the lineage. It can turn the snail into a release point for very large numbers of infective larvae. For eye flukes such as Diplostomum, that matters because open water is a difficult place in which to find fish one by one. The parasite solves that problem with numbers. It does not trust a single attempt. It produces many. This is not the charming side of biology, but it is the effective side, and parasites tend to favour that. When the cercaria leaves the snail, the parasite enters one of the shortest and most exposed parts of its life. It is briefly out in the water, free swimming but not free in any meaningful sense. It has a job and very little time to do it. Studies on Diplostomum spathicium sacariae have shown that transmission success depends strongly on survival time and rapid contact with a fish host. Once a fish is encountered, the Sacaria penetrates through the skin or gills rather than entering directly through the eye. After that, it migrates internally, often reaching the eye lens within 24 hours. That detail is important because it tells us how the parasite actually lives during infection. It is not drifting into the wrong place. It is moving through a fish with remarkable purpose, using the body like a route rather than merely a barrier. And then we come to the part that gives the animal its common reputation. In many of the best-known eye flukes, particularly Diplostomum pseudospathicium, the larva reaches the eye lens and settles there as a metacercaria. Researchers describe the fish eye lens as an immunologically privileged or inert site, which means the parasite gains a degree of protection from the host's adaptive immune response once it arrives. That is one reason the lens is such a useful place for the parasite to live. It is not just damaging, it is sheltered. The fluke is not merely hiding from the fish in the ordinary sense, it is occupying a tissue where the host's ability to attack it is reduced. From a parasite's point of view, this is excellent housing. From a fish's point of view, it is an appalling arrangement. The word live is also worth slowing down for here, because the metacercaria in the eye is not living like an adult bird stage worm lives in an intestine. It is not there to reproduce sexually, it is there to persist, grow to the next ready state, and wait for transfer. Some studies describe eye lens metacercariae continuing to grow and develop in the lens over time, with temperature influencing their development rate. This waiting stage can last until the infected fish is eaten by a suitable bird. So the eye fluke's life in the eye is not simply residence, it is residence with a deadline attached. It lives there long enough for the fish to become part of a bird's meal, because only then can the parasite reach adulthood. That makes the fish eye a staging area rather than a final home, though admittedly it is a staging area with very poor reviews from previous occupants. Not all eye flukes live in exactly the same part of the eye, and this is where the subject becomes more varied than the phrase eye fluke suggests. Species of Diplostomum and related diplostomids have been recorded in the lens, the vitreous humour, the retina, and the subretinal space. A recent study on wild sticklebacks described eye fluke larvae invading the subretinal space, causing retinal detachments and damaging the retina and retinal pigment epithelium. Other summaries note that fish eye flukes can form diverse communities within fish eyes rather than occupying one uniform niche. So when people imagine one standard parasite sitting identically in every fish eye, the real picture is messier. Some species behave as classic lens flukes, others use different eye tissues. The result is the same broad idea, but the details of damage and pathology can differ quite a bit. This is also why the taxonomy of eye flukes has been trickier than older literature sometimes made it seem. Modern molecular work has shown that the diversity of Diplostomum in natural fish populations is greater than earlier morphology-only identifications often suggested. Studies from South Africa and elsewhere have used molecular and morphological approaches together to sort out species boundaries and host associations more reliably. That matters because different species may use different snails, different fish, and different eye tissues, even when they look annoyingly similar under a basic identification scheme. To say eye fluke is therefore useful in the way saying songbird is useful. It tells you the broad sort of thing you're dealing with, but not the full identity. The parasite itself, meanwhile, continues being indifferent to our naming difficulties and gets on with infecting fish. How they live inside the host is partly a matter of feeding, partly a matter of simply existing in the right place. Trematodes generally are adapted for parasitism through a specialized body surface and physiology suited to nutrient uptake, host invasion, and survival within other animals. Science Direct's overview describes trematodes as highly adapted parasites whose physiology has been shaped around invasion, modulation of host physiology, nutrient intake, and successful reproduction. In the fish eye, the metacircarial stage is not roaming about like a tiny predator. It is lodged in tissue, interacting with its surroundings, causing mechanical and physiological disruption, and in many cases contributing to cataract formation as parasite numbers increase. That is an important distinction. An eye fluke does not need to behave dramatically to have a large effect. It only needs to be present in the wrong place in sufficient numbers. The severity of that effect depends heavily on burden. Research on Diplostomum spathicium found that cataract intensity rises significantly with the number of parasites established in the lens, and later summaries note that heavy infections can involve very large numbers of metacircariae. High parasite burdens are associated with more severe lens opacity, poorer vision, and in some cases acute mortality or strong sublethal effects. So an eye fluke does not live in the eye as a harmless occupant that merely happens to be there. It lives there as a parasite whose very presence changes the optical quality of the lens and can compromise the fish's performance in ordinary tasks such as feeding, schooling, and predator avoidance. For a fish, that means the difference between a manageable world and an increasingly blurred one. There is another practical point hiding inside all of this. Eye flukes are not trying to make fish sick for the sake of making fish sick. They're trying to complete a trophic transfer. That means the way they live in the fish has to balance persistence with transmission. If they destroy the host too fast, they may never reach a bird. If they do too little, the fish may remain too competent and too hard for a bird to catch. This is why the eye is such an efficient sight. Vision impairment can increase predation risk without immediately ending the host's life. Studies on anti-predator behaviour and natural predation have shown that infected fish can become more susceptible to bird predation, which is exactly what the parasite needs. In that sense, the eye fluke lives as both a passenger and a quiet engineer of worse odds. So when we say what eye flukes really are, the simplest honest answer is this. They are parasitic flatworms, usually diplostomid trematodes in this context, whose life cycle is divided among birds, snails and fish, and whose larval stages are specialised to live in fish eyes, especially the lens, where they can survive, develop and alter the host's vision until the fish is eaten by a bird. They are not monsters in the theatrical sense. They are specialists. Their bodies are simple, their life cycle is complicated, and their success comes from matching each form to each host with very little waste. That is how they live. Not grandly, not loudly, just effectively enough to keep moving from one body to the next. And once you understand them in that practical way, the next thing that becomes easier to follow is the damage they leave behind. Because the eye fluke story is not only about what the parasite is, but about what happens to vision when something this small takes up residence exactly where clarity matters most. Underwater vision is already a demanding thing even before a parasite interferes with it. A fish is not looking through clear empty air. It is reading a world of filtered light, suspended particles, moving reflections, shifting depth, and contrast that can vanish almost as quickly as it appears. For many fish, vision is still a dominant sense used for foraging, predator avoidance, social behaviour and orientation, which means damage to the eye does not remain a local problem for long. It becomes a whole body problem. One study on wild stickleback states this very plainly in its opening lines. Fish rely on vision as a dominant sensory system for foraging, predator avoidance and mate selection. When eye flukes damage the visual system, they are not merely clouding an organ. They are interfering with one of the fish's main ways of understanding where food is, where danger is, and where it ought to be in relation to everything else around it. The most familiar damage caused by classic eye flukes is cataract formation. In fish infected with Diplostomum species that establish in the lens gradually becomes cloudy as parasite burden increases, and that clouding can be measured and scored. A quantitative study in rainbow trout found that cataract intensity rose significantly with the number of Diplostomum spathicium parasites established in the lens. That is useful because it gives us something more precise than a vague sense of bad eyesight. The more parasites successfully settle in the lens, the more the lens loses the transparency it needs in order to focus light cleanly onto the retina. In practical terms, the fish does not simply see less. It sees less sharply, less reliably, and with a steadily narrowing margin for error. A fish that once picked out prey with a quick correction of the head may now misjudge distance, miss the target, or react one fraction of a second too late. That one change, the loss of lens clarity, has a way of spreading into everything else the fish does. Infected fish may still swim, feed, and school, but the quality of those actions changes because the visual information feeding them becomes less trustworthy. A review of eye fluke effects in round gobies notes that in several fish species, impaired vision due to eye fluke infection has been shown to reduce feeding capability, increase feeding times, and raise predation risk. The wording there matters. Reduce feeding capability, increase feeding times. Those are not dramatic theatrical consequences. They are subtle degradations of competence. The fish can still attempt to live normally, but it does so more slowly and less effectively. Another study on European perch found that eye fluke infection was associated with changes in diet composition, in a context where the authors note that eye flukes impair host vision and have been linked to altered feeding behaviour. That suggests the fish is not merely eating less. It may begin eating differently because the prey it can detect and capture efficiently is no longer the same. There is something especially harsh about that underwater, because prey detection below the surface is often a matter of quick visual decisions made in imperfect conditions. The fish is not browsing under gallery lighting. It is working with dimmer light, changing angles, surface glare, turbidity, vegetation, shadows, and motion. In that setting, even mild cataracts can matter more than they would in a clean laboratory fantasy. A tiny moving invertebrate, a drifting zooplankton target, the edge of a shelter, the break in a weed line. All of these may depend on the fish being able to separate contrast from noise. Cataracts do not have to cause complete blindness to be costly. They only have to reduce that contrast enough to turn a successful strike into a miss, or a timely retreat into a hesitation. Research in farmed rainbow trout found that cataract severity correlated strongly and negatively with later body mass and condition, which is exactly the kind of downstream effect you would expect if fish are consistently feeding less effectively over time. Predator detection may be even more important than feeding when the water above the fish is full of birds. Eye flukes benefit when infected fish become easier prey for avian hosts, and several studies support the idea that cataract severity is linked to weaker anti-predator performance. In round gobies from the Baltic, more intense parasite-induced cataracts were associated with weaker responses to simulated avian attack, even though some other anti-predator behaviours such as shelter use and shade preference were not clearly impaired. In earlier work with rainbow trout, infected fish showed decreased escape responses and increased susceptibility to artificial aerial predators. That is a bleakly elegant arrangement. The fish does not need to forget all fear or stop avoiding cover entirely. It only needs to become slightly worse at reading the fast top-down danger of a bird strike. In the life cycle of an eye fluke, that small reduction in precision may be enough. And of course that danger arrives from above, which makes the visual challenge even more specific. A fish detecting an avian predator is often trying to read a silhouette, a sudden movement, a changing shape against the brightness of the surface or the sky beyond it. Cataracts blur exactly the kind of crisp information needed for that job. The fish may still register brightness and motion, but the timing of its reaction can worsen. One study on yellow perch infected by Diplostomum bari did more than infer this from behaviour. It measured retinal function directly using electroretinograms and found that infected fish had a reduced B-wave amplitude, increased latency, and lower flicker fusion frequency. In plain language, the retina's physiological response to light was measurably impaired. That means the parasite's effect is not just an outward behavioural story we are projecting backward. In at least some host-parasite combinations, the visual system itself, all the way down to light response is functioning less well. That yellow perch work is especially important because it pushes the discussion beyond cataracts alone. Many people hear eye fluke and imagine a parasite sitting in the lens like a grain of bad luck turning the whole thing cloudy. That is one major pattern, but it is not the only one. In yellow perch, Diplostomum beri was found in the choroidal vasculature rather than the lens, and the adjacent choroid, retinal pigment epithelium, and retina showed extensive damage. In wild three-spine sticklebacks, another study found eye flukes frequently positioned between the neural retina and the retinal pigment epithelium, with clear tissue damage and even fragments of retinal pigment epithelium observed within fluke bodies, suggesting the parasites had consumed eye tissue. So beneath the water surface, the visual cost may come from two different directions at once. Light may fail to pass cleanly through a clouded lens, and the tissues meant to process that light may themselves be physically damaged. That combination is worse than it first sounds. A cloudy lens is like looking through frosted glass. A damaged retina is like having the image sensor itself partly disrupted. If both are compromised, the fish is not merely struggling with blur. It may be dealing with reduced sensitivity, poorer resolution, slower visual processing, and in some cases structural injury that changes how retinal layers line up and respond. The stickleback study described laminar fusions, in which neuronal cell bodies were misplaced within a major synaptic layer of the retina, a pattern consistent with retinal injury and regenerative activity. The yellow perch study showed that retinal light responses were measurably altered. Taken together, these findings make it clear that eye flukes can affect vision at more than one level. They can interfere with the incoming optics and with the neural tissues that interpret the signal. For a fish trying to live in a world of motion and threat, that is not a minor inconvenience. It is the equivalent of being forced to navigate with a fogged window and a damaged dashboard at the same time. Feeding becomes especially revealing once vision starts to fail, because feeding is one of the first places where poor sight turns into poor condition. A fish with healthy vision can make quick, cost-effective decisions: strike, seize, swallow, move on. A fish with cataracts or retinal damage may miss more often, spend longer searching, and waste energy on unsuccessful attempts. The Round Goby paper notes that infected fish in the field had poorer condition, and that this may reflect impaired feeding due to eye fluke-induced cataracts, consistent with results seen in other species. Earlier work on Arctic char suggested that cataract-bearing fish spent more time foraging to compensate for energetic costs and reduced feeding capability. That creates a miserable trade-off. The fish has to expose itself more, move more, and spend more energy just to secure the same nutritional return, all while its weakened vision makes each feeding attempt less reliable, and each extra minute in the open more dangerous. It is also worth noticing that underwater vision is not only about seeing objects, it is about being able to keep up with a moving environment. Fish use visual information to hold position relative to current, maintain spacing in a school, choose background, and decide when to freeze or dart. One review-style paper on eye flukes and anti-predator behaviour notes that infected rainbow trout show reduced preference for dark background coloration and impaired ability to adjust coloration to the background, which can make them more conspicuous. That matters because crypsis is partly a visual task and partly a visual outcome. The fish must both choose the right background and match it effectively. If its visual input degrades, concealment itself may become worse, so the effect of eye flukes beneath the surface is not confined to what the fish sees. It also changes how the fish is seen. A clouded lens can eventually turn into a more visible body. At the same time, the story is not quite as simple as worse vision explains everything. Some newer work has argued for a more careful interpretation. A 2023 study on eye fluke-infected fish tested behavioural change under different lighting conditions and found that the difference between infected and control fish was independent of those conditions, suggesting that mechanisms other than vision impairment should also be considered in this system. That is a useful bit of scientific restraint. It reminds us that parasites can change host behaviour through multiple routes, including pathology, inflammation, stress, altered energy budgets, or other tissue-level effects, rather than through eyesight alone. So when we imagine an infected fish moving badly beneath the surface, it is safest to think of degraded vision as a major factor, often a central one, but not always the whole explanation. Biology is usually less tidy than our first narrative draft. Even so, vision remains one of the strongest and most consistent links between eye fluke infection and ecological consequence. The evidence for cataract formation, retinal injury, reduced retinal function, feeding impairment, weaker escape responses, and increased vulnerability to avian predators points in the same general direction. The parasite alters the fish's visual world enough that normal underwater life becomes harder to perform. USGS work on endangered suckers in Upper Klamath Lake summarises the endpoint in brutally simple terms. The fish are blinded by the eye fluke and then eaten by water-faring birds. That wording is a little plainer than academic phrasing, but sometimes plainness is useful. It strips the process down to the central fact. Beneath the surface, the fish is losing the ability to read the world above it, around it, and in front of it. Once that loss becomes severe enough, the rest of the life cycle takes over. What makes this especially effective for the parasite is that the damage does not have to be all or nothing. There is no requirement that every infected fish go blind in a dramatic instant. A gradient of harm is often enough. The quantitative cataract study showed a burden-dependent relationship, which means the parasite can create a sliding scale of impairment rather than one fixed outcome. Some fish may carry mild infections and function reasonably well. Others may accumulate enough metacercariae that the lens grows heavily opaque. In natural populations, infection intensities and species combinations vary, so the result is not one single visual syndrome but a range of underwater difficulties, from subtle reduction in acuity to major clouding and retinal compromise. For ecology, that is important, because it means the parasite can shape host behaviour and survival probabilities across a population, not only in the worst visible cases. There is also a cruel efficiency in the timing. The fish is often infected while young, and still developing in habitats where snails release cercariae in large numbers. If visual damage begins early, the fish may spend a long part of its life paying the cost through poorer feeding and greater exposure. In Klamath Basin research, scientists are studying juvenile endangered suckers to understand the concentration of eye flukes that can directly kill them or make them highly susceptible to bird predation. That focus on juveniles tells its own story. A young fish already has a difficult job. It needs to grow, avoid being eaten, and make enough correct decisions in a visually messy environment to reach maturity. If parasites compromise vision at that stage, the cost is multiplied over the fish's whole early life rather than imposed at the very end. When you imagine all of this from the fish's perspective, the effect is less dramatic than a horror image and more relentless than one. The water does not suddenly become black, it becomes less readable. Prey flickers by and is harder to judge. Shadows from above become more ambiguous until the instant they are not. A schoolmate turns and the spacing is harder to maintain. A patch of safer background no longer draws the same reliable visual cue. The fish may spend longer foraging, rise more often into risky space, react later to a strike from above, and emerge from all of it with poorer condition. None of those moments would look spectacular in isolation. Together they shape a life with narrower margins. This is how many parasites do their best work. They do not always kill with a flourish. They make ordinary success less likely, over and over again, until chance begins to lean the wrong way. That is why the phrase effect vision is almost too small for what is happening. The eye fluke changes more than eyesight. It changes the way the fish can feed, hide, react, orient, and remain inconspicuous in a medium where small visual advantages often decide survival. It can cloud the lens, damage the retina, slow light responses, reduce feeding efficiency, worsen body condition, and weaken escape from aerial attack. Some behaviours may shift for reasons beyond vision alone, but vision still sits at the centre of the disturbance in many of the best studied systems. Beneath the water's surface, the fish is not merely seeing a blurrier world, it is living in a more dangerous one, and the parasite's entire route through snail, fish and bird depends on that danger becoming just a little easier to realise. And once you start looking at the damage in that practical way, the next thing that matters is where this keeps happening, because eye flukes are not scattered randomly through every body of water in equal fashion, and the fish that carry them tend to come from the kinds of places where snails, birds, shallow margins and repeating exposure all meet. By the time an eye fluke reaches a fish, it has already made one important demand of the landscape. It needs the kind of water where hosts do not merely exist but overlap. That usually means fresh water first, and very often the quieter parts of it. Diplostomum species are widely distributed parasites of freshwater fish around the world, though related eye flukes also occur in brackish systems, and the literature repeatedly describes them as common in freshwater and brackish water fishes rather than creatures limited to one dramatic marine setting. One review summary describes Diplostomum as generally located in the lens of freshwater fish worldwide, while another notes that Diplostomum species infect freshwater and brackish water fishes and can occupy different parts of the eye. This matters because it places the parasite in ordinary waters, lakes, ponds, reservoirs, marsh edges, slow channels, sheltered bays, and mixed coastal zones where birds feed, snails graze, and fish spend enough time in contact with both to keep the cycle going. The fish that carry them are not one neat club of doomed specialists. Eye flukes use a broad range of second intermediate hosts, and some lineages are distinctly flexible about which fish species they will enter. A review of environmental variation in host-parasite interactions describes Diplostomum pseudospathicium as having a broad range of fish host species, and another host diversity study found roughly 5,000 Diplostomum metacercari across multiple freshwater fishes, with particularly high abundances in yellow perch, blunt-nosed minnow and golden shiner, and lower but still detectable abundances in pumpkin, seed, rock bass, Johnny darter, and yellow perch in other eye sites. In Europe, juvenile European perch are described as serving as intermediate hosts to a diverse number of eye fluke species, and the authors note that eye fluke infections are highly prevalent in perch populations across Europe. So the fish carrying these parasites are not only one familiar victim such as trout or one unlucky lake species, they include minnows, perch, gobies, sticklebacks, cyprinids, and other freshwater fishes that happen to live where the snail-to-fish step is easy to repeat. Three-spined sticklebacks are a very good example of the kind of fish that often end up in the middle of this arrangement. The Münster research summary on Dios pseudospathicium states that the parasite is frequent in many freshwater fish species, including sticklebacks, particularly in lakes with limnaeid snails, which are the first intermediate hosts. That one sentence explains a great deal. It is not simply that sticklebacks are susceptible, it is that their habitat can place them exactly where the parasite needs them to be. Lakes with limnaeid snails offer repeated exposure, and the same study notes that cercariae are shed from late spring to autumn, giving fish repeated contact over the warmer part of the year. A lake stickleback is therefore not merely carrying a parasite because of bad personal luck. It is occupying a setting where shallow water, snail abundance, and seasonal release all work in the parasite's favour. The fish is small, common, often near productive edges, and constantly moving through the very zone where infective larvae are most likely to be waiting. European perch tell a slightly different version of the same story. They are visual predators, and one of the most abundant fish species in northern temperate lakes, which already makes them useful hosts from the parasite's point of view. The scientific report study on juvenile perch sampled fish from temperate Lake Müggelsee near Berlin and found not only eye fluke infections, but diet changes associated with increasing infection intensity. More importantly for this part of the story, the paper notes that perch are widely distributed across the Palearctic and commonly host multiple eye fluke species. That is exactly the sort of fish a parasite can build a reliable route around. Abundant, lake-dwelling, visually dependent, and present in waters where snails, zooplankton, benthic prey, and fish-eating birds already interact at close range. A fish does not need to be rare to matter in a parasite's life cycle. In fact, common fish are often the better bet. The parasite's ideal host is not the most glamorous fish in the water. It is the one most available in the right place, year after year, while birds continue taking meals from above. Then there is the round goby, which proves that the waters favoured by eye flukes are not only inland lakes and ponds. In Kalmar Sound in the Baltic Sea, a brackish water system, round gobies were found with eye fluke prevalences of 90 to 100% in the Flink, Behrens and Svensson study. The fish in that work were collected in shallow water, 0 to 1 metre deep, and held originally in continuously flowing brackish water of about 7 parts per thousand. The same paper points out that multiple native eye fluke species can co-infect round gobies in the Baltic region, and that important avian predators there include great cormorants and grey herons. This is a useful reminder that the parasite does not insist on one specific postcard setting. Give it snails, fish and birds in a connected brackish habitat, and it will manage quite well. The water can be fresh, lightly salty or somewhere in between, provided the route remains intact. Shallow coastal margins, estuarine-like brackish zones, and sheltered nearshore habitats can support the same unpleasant choreography as an inland lake. Trout add another piece to the picture, especially where people have altered water flow or concentrated fish. Science Direct's overview on fish disease describes dyspathseum as transmitted by seagulls and parasitizing rainbow trout during summer months, with larvae migrating to the eye and causing cataracts, blindness, inflammation, and hemorrhagic injury. The same source notes that controlling the disease often comes down to breaking the life cycle by targeting snails, especially in ponds or channels where weed growth has occurred, because the snails prefer slow-moving water with vegetation on which to live and feed. That single habitat note is very revealing. It tells you what kind of water eye flukes favour, even when no one is using poetic language about it. They like the ordinary, productive, slightly weedy, slow places, not roaring torrents, not sterile concrete basins scrubbed of life. The parasite prospers where snails can stay put, browse, and keep shedding larvae into water repeatedly visited by fish. Trout farms, hatchery channels, pond margins, and sheltered inlets can all become unusually useful to the parasite if those conditions line up. The fish themselves also differ in where the parasites settle once they arrive, and that can shape which hosts end up carrying heavy burdens. Lock's diversity study showed that among 6 intensively sampled freshwater hosts, Diplostomum metacercariae were most abundant in the vitreous humour of yellow perch and in the lenses of blunt-nosed minnow and golden shiner. They were much less abundant in several other host site combinations. That is worth noticing because it means not every fish is equally good housing for every eye fluke species. Some fish are simply better matches. A broad host range does not mean complete uniformity. It means the parasite has options, and some of those options are especially productive. A minnow in one lake, a shiner in another, a perch in a mixed littoral zone, a goby in brackish shallows, a stickleback in a snail-rich lake. The parasite keeps finding bodies that fit well enough, and those repeated good enough matches are exactly what sustain a widespread parasite over large geographic areas. The waters they favour are therefore less mysterious than they first sound. They are waters with structure, vegetation, shallows, productive margins, snail habitat, repeated fish use, bird access. The old aquaculture guidance in the Science Direct overview puts it bluntly by noting that the snails prefer slow-moving water with some vegetation and that ponds or channels with weed growth should be cleared if managers want to reduce Digenean infections. The Münster summary adds the seasonal piece by noting repeated cercarial shedding from late spring to autumn. Together, those details describe the sort of water where eye flukes do best. Not fast, barren water that sweeps everything away, but slower and more settled places where hosts meet often. Literal zones of lakes, marshy edges, reedy embayments, shallow estuarine margins, calm backwaters, and nutrient-rich systems all start to look less peaceful. Once you realise that from a parasite's perspective, they are not just habitat, they are opportunity arranged into geography. Upper Klamath Lake is a good real-world example of how much the surrounding ecosystem matters. USGS researchers studying endangered suckers there describe warming temperatures, habitat alteration, and nutrient inputs as part of an altered ecosystem that may be increasing snail abundance, and they note that more snails may mean more eye fluke parasites. In a separate USGS summary, they also point out that highly nutrient-rich waters could increase snail abundance, while more transparent waters or more birds could raise predation on infected fish. This is exactly the sort of practical ecology that turns a parasite from an odd laboratory subject into a conservation concern. The fish carrying these parasites in Klamath are not random experimental animals. They are juvenile lost river and short-nosed suckers living in a lake where environmental conditions may be making the life cycle easier to complete. The parasite favours not only certain waters then, but waters that have become especially good at producing its first intermediate host and exposing young fish to repeated contact. That helps explain why younger fish can matter so much. Juvenile fish are often concentrated in shallow nursery areas, feeding in precisely the places where snails are abundant and cercariae are being released. The perch study focused on young-of-the-year fish in a temperate lake. The Klamath work focuses on juvenile suckers. The rainbow trout literature often discusses fry or young fish in rearing settings. This is not accidental. Smaller fish spend more time in risky margins, have less room for anatomical error, and may accumulate infections early, before they have grown out of the shallowest habitats or developed whatever resistance their species can mount. Some fish carry eye flukes simply because they are the fish most exposed during the most vulnerable stage of life. A juvenile fish in a warm, shallow, snail-rich summer habitat is almost serving the parasite an address card. It does not intend to, of course. It is simply trying to grow in the part of the water most suited to feeding, and that happens to be the same part of the water most suited to parasite transmission. There is also a geographic point worth making quietly. These parasites are not a local curiosity confined to one famous lake or one region's field guide. The host and habitat records span North America, Europe, Africa, and brackish parts of the Baltic. The South African work summarised in the search results recovered Diplostomum metacercaria from the eye lenses of 38 fishes belonging to 5 species from several families, showing that the pattern is not locked inside a narrow northern story. A separate review summary describes Diplostomum as common parasites of freshwater fish worldwide. The exact fish differ from place to place, the snail species can change, and the final bird host may vary, but the broad arrangement remains familiar. If a body of water supports the right snails, fish, and fish-eating birds, the parasite has a chance. The specific cast can change. The script stays recognisable. This is one reason eye flukes can be so successful in invaded or changing systems as well. The round goby study notes that the eye fluke species infecting gobies in Kalmar Sound were native to the Baltic region rather than introduced with the goby, which suggests that a newly abundant fish can be folded into an existing parasite network if it proves to be a suitable intermediate host. That is a very useful ecological lesson. Parasites do not always need their traditional fish forever. Sometimes they only need a fish that occupies the right space in the right water and can be eaten by the right birds. In a changed ecosystem, the cast can be updated without the root falling apart. A bold invasive fish may discover that local life has already prepared a quiet welcome committee, and the welcome committee consists largely of snails and larval flatworms. So the fish that carry eye flukes tend to be the fish that live where the parasite's three-host route becomes routine rather than rare. Lake sticklebacks in snail-rich systems, perch in temperate lakes, minnows and shiners in productive freshwater communities, trout in slow, vegetated waters and managed channels where snails can thrive, round gobies in shallow Baltic brackish habitats, juvenile suckers in nutrient-rich lake systems where snail numbers may be rising. The waters they favour are not defined by romance or menace. They are defined by overlap. Slow enough for snails, busy enough for fish, open enough for birds, and stable enough for the cycle to repeat. Once researchers began noticing that pattern across so many places and hosts, the story of eye flukes stopped looking like a string of isolated oddities and started looking like a consistent feature of aquatic life, which is exactly where the science becomes even more interesting, because the next step was to figure out how anyone realised what these tiny passengers were doing in the first place. Scientists did not uncover the secret life of eye flukes in one dramatic moment, with one brilliant person peering into one microscope and instantly understanding everything. The discovery came in pieces, over a long stretch of work, because the parasite itself lives in pieces. One form appears in a snail, another in a fish, another in a bird, and none of those stages fully explains the others unless someone has the patience to connect them. A modern review of parasite life cycle studies makes that point very clearly. The old parasitological tradition of elucidating full life cycles was built around identifying all the hosts used by a parasite at its different stages, precisely because looking at only one stage gives an incomplete answer. Eye flukes are a perfect example of why that work mattered so much. You can find something in a fish eye, name it, and still not know where it came from or where it's going next. The earliest part of the story was therefore a taxonomic one. Parasitologists first had to realise that the tiny worms found in different hosts might belong to the same broader lineage at different points in life. The genus Diplostomum dates back to von Nordmann in 1832, and modern systematic papers still refer to it that way because the historical naming matters when later researchers try to untangle old descriptions from modern species concepts. By the early 20th century, names such as Diplostomum foxini had entered the literature as well, but the problem was never just naming the worms. The problem was that larval stages in fish and snails often look different enough from adult stages in birds that, without life cycle work, they can seem like separate puzzles rather than one organism moving through time. That is one reason the group accumulated a long and somewhat difficult taxonomic history before modern tools began to clean it up. Before DNA sequencing, the basic way into that puzzle was careful watching. Scientists dissected fish, examined cloudy lenses, looked for metacocaria in eye tissues, checked snails for shedding cocaria, and searched the intestines of fish-eating birds for adult worms. None of that sounds glamorous now, but it was the core method. Trematode life cycles were built by linking bodies, tissues, and habitats through direct observation and experimental infection. The 2017 Cambridge Review on Life Cycle Studies argues that this kind of work used to occupy a much more central place in parasitology than it does now, precisely because it required the researcher to move across hosts rather than staying in one convenient part of the system. With eye flukes, that old method was especially important because the fish stage alone can tell you a great deal about damage, but almost nothing about where the parasite began or how it reproduces. One of the important mid-century to late 20th century steps was simply to show that the common fish eye fluke was not an isolated curiosity in wild fish, but something that could be studied under both natural and experimental conditions. R. A. Sweeting's 1974 paper, Investigations into Natural and Experimental Infections of Freshwater Fish by the common eye fluke Diplostomum spathaceum, has remained a major reference point for later work, which tells you how foundational that stage of the research was. Scientists were no longer only cataloguing strange parasites found by chance. They were beginning to expose fish under controlled conditions, compare natural and experimental infections, and ask practical questions about infectivity, pathology, host exposure, and disease significance. Once that happened, flukes moved from the margins of descriptive zoology into a more active kind of fish parasite science. From there, the work became more ambitious. Instead of asking only whether fish could be infected, researchers started trying to complete and maintain full life cycles in the laboratory. A 1995 study by McKeown and colleagues stated its objective directly: to establish and maintain the life cycles of several Diplostomum species in the laboratory so that accurate and detailed descriptions of all stages could be produced. That kind of sentence may not sound thrilling at first glance, but it marks a very important shift. Once you can run the parasite from one host stage to the next under controlled conditions, you stop guessing about connections and start demonstrating them. You can say which Cercaria becomes which metacercaria becomes which adult, and what features remain stable or change along the way. For a parasite whose identity had long been blurred by stage-specific forms and difficult morphology, that was a major step forward. Even then, however, the microscope was not enough. That is one of the recurring themes in the modern literature on Diplostomum. Morphology mattered, and still matters, but larval flukes are often difficult to identify to species by appearance alone. A large 2015 review on larval Diplostomidae explains that eye-dwelling diplostomids are frequent subjects of ecological and evolutionary study, yet their metacercariae are difficult to identify morphologically, which is why molecular methods became so important. The same paper notes that sequences from adult worms are usually needed if researchers want to identify species rather than merely distinguish unnamed lineages. In other words, scientists had discovered much of the visible life cycle, but they still faced a problem familiar to anyone who has tried to sort a drawer of nearly identical keys. They knew they had multiple species. The question was, which was which? That is where the molecular era changed the field. By the early 2000s, and especially the 2010s, parasitologists increasingly began linking larval and adult stages using ribosomal and mitochondrial DNA markers. Brabeck and colleagues summarised the problem sharply in 2015, writing that the model systems used in many ecological and pathological studies had often been referred to as Diplostomum spathaceum in a collective sense, or as unnamed Diplostomum species, because accurate species identification was lacking. They emphasised that identification of the larval stages is particularly difficult since linking life cycle stages requires experimental completion of the life cycle. Their solution was part of a larger shift toward molecular baselines, complete mitochondrial genomes, and ribosomal markers that could support barcoding and epidemiological work from the most accessible life cycle stages. That was not just a technical upgrade, it was a way of turning a historically blurry parasite group into something much more traceable. A particularly important milestone came in 2013, with molecular prospecting for European Diplostomum reveals cryptic diversity. The title alone tells the story. Scientists were no longer just confirming a few familiar species, they were prospecting, which is exactly the right word for a field where the diversity had been underestimated for so long. The authors described the study as the first attempt to address molecular prospecting for species diversity of Diplostomum in Europe, and later papers repeatedly cite it as the moment when hidden diversity became impossible to ignore. Once researchers began sequencing across larval and adult stages, the neat older picture of one or two common flukes started to fracture into multiple lineages, species complexes, and provisional species that had been concealed inside morphology-based categories. It turned out that the fisheye had not been hosting one familiar enemy. It had often been hosting several. That discovery changed how scientists read older literature. If previous work had pulled many infections under broad labels, then some earlier conclusions about host range, pathology, seasonality and geography might have been blending several species together. The 2015 review on larval Diplostomidae makes exactly that point when it notes that DNA sequences from Diplostomids are accumulating worldwide. that molecular methods are often used to distinguish species, and that provisional species delineations have become valuable because ecology can look different depending on whether infections are pooled into one genus-level category or resolved into distinct species. This was not a mere academic naming dispute. It meant that the common eye fluke of older fish pathology could actually be several closely related parasites with different host preferences, tissue sites, or ecological patterns. Once that possibility became clear, the secret life of eye flukes looked not only more complex but more crowded. At the same time, scientists discovered that sequencing alone could not solve everything. A 2022 paper on best practice in Diplostomum systematics warned that DNA data had become integral to species characterization and identification, but also stressed that specimens associated with the sequence still have to be identified through traditional morphology and correct life cycle linking. The authors also pointed out a striking limitation in the available databases. Less than 6% of the DNA sequence data then available for Diplostomum originated from adult specimens, even though adult isolates are essential for accurate identification. That is an unusually honest reminder that modern parasitology did not replace the old tradition so much as become dependent on it. The sequence is powerful, but only if someone can say which actual worm it came from, in which host, and how that stage connects to the others. The microscope did not lose, it simply had to learn to share the table with genetics. Some of the most elegant recent studies are the ones that deliberately reunite those approaches. In 2021, Schwelm and colleagues characterized Diplostomum foxini using both morphology and molecular data, basing the identification on cercarial isolates from a limnaeid snail and metacercaria from the Eurasian minnow, and they provided molecular evidence for the identification of the snail intermediate host. The same paper reported phylogenetic analyses that depicted 44 molecularly characterized species and genetically distinct lineages of Diplostomum. That is the sort of sentence that would have been unimaginable in the older period when fisheye larvae were often hard to distinguish by shape alone. It also captures what scientists had really discovered by that point. Not just the life cycle of one parasite, but an entire hidden community of related parasites moving through snails, fish, and birds under names that had only recently begun to catch up with reality. Another useful example comes from North America. A 2016 study on Ostrodiplostomum ostrowskii, another fisheye fluke in the Diplostomid family, reported that the researchers molecularly linked the Cercaria, metacercaria, an adult stage of the life cycle, and identified the snail host for the parasite. That may sound like a very specialized result, but it shows the mature form of the method scientists now use to uncover these parasites' private roots. They do not just collect one odd worm from one odd place. They connect the snail stage, the fish stage, and the bird stage with direct evidence. Similar work in Brazil combined morphology, experimental infections, and molecular data to identify several diplostomoid cercaria from snails, and the metacercaria obtained after exposing laboratory-reared fish. In other words, the discovery process now often involves staging the parasite's own journey under controlled conditions and checking every stop with DNA as well as anatomy. As the tools improved, scientists also stopped looking only at single parasites and began looking at communities. In 2011, Rellstab and colleagues developed a pyrosequencing approach to analyze trematode parasite communities in fish eye lenses using naturally pooled DNA. Their study showed that multiple species infections were common and that different fish species harbored significantly different parasite communities. This was an important conceptual shift. The secret life of eye flukes was no longer just about connecting one snail, one fish, and one bird. It was about recognizing that a single fish eye can contain a community of closely related parasites, some of them difficult to separate morphologically, all interacting within the same host. The eye had gone from being a place where a parasite was found to a place where parasite diversity itself could be measured. By 2024, that community-level approach had advanced again with targeted DNA metabarcoding. A study on fisheye fluke communities in Estonian lakes developed a cost-effective COX-1 region-specific metabarcoding method and applied it to 2 common fish species across 7 temperate lakes. The researchers found substantial interlake, intralake and inter-host variation in diplostomid communities, as well as positive and negative associations among co-infecting species. The significance of that result goes beyond method. Scientists had moved from the era of what is this worm in this fisheye to how do entire eye fluke communities vary across space, season, host species and environmental context. That is not merely a better lens, it is a different scale of understanding altogether. And yet the older methods remain visible beneath the newer ones. Even the most modern papers keep circling back to the same practical difficulties. Larvae in fish are hard to identify. Adults in birds are essential for naming. Snail hosts must be confirmed, and experimental completion of the life cycle is often needed to make the final connection. The 2022 best practice paper and the 2017 review on life cycle studies both, in different ways, make the same argument. There is no shortcut that fully replaces host-by-host evidence. The hidden life of a trematode cannot be discovered by sequencing alone if nobody knows which body the sequence came from. Nor can it be solved by morphology alone if the species are too similar in the stage most often collected. What scientists discovered then was not just the parasite's route, but the need for a mixed method equal to that route. Dissection, microscopy, experimental infection, histology, host ecology, and molecular linking all working together. There is a quiet irony in all of this. The parasite itself is very small, but the work required to know it properly is very large. To understand an eye fluke, scientists have had to look into fish eyes, collect snails, rear hosts, examine bird intestines, compare old species descriptions, build phylogenies, and keep correcting names as new evidence arrives. What looks from a distance like one unpleasant little worm has turned out to be an entire scientific exercise in patience. That is probably why the story of discovery feels so fitting for the parasite itself. Eye flukes live by connecting separate hosts into one cycle, and scientists learned their secret life by doing much the same thing with separate observations until they formed one coherent picture. The cloudy lens in a fish, the fork-tailed cercaria in a snail, the adult worm in a gull, the DNA sequence in a lab, each by itself was only part of the truth. Only when they were brought together did the life cycle begin to speak clearly. That clearer picture, however, did not make the story simpler. It made it more intimate, because once scientists understood the route and the identities involved, the next question became harder to avoid. If the parasite is so precisely adapted to the fish eye, and if fish are not merely passive containers but living hosts with defences of their own, then what exactly happens in that struggle between invasion and resistance once the fluke arrives and the host begins, however imperfectly, to answer back? What makes the struggle between an eye fluke and its host so unsettling is that it is both fierce and brief. We tend to imagine a host fighting continuously, as though the immune system stands in full armour at every doorway, ready to stop an invader at leisure. That is not quite how this parasite works. In lens-infecting Diplostomum species, the Cercaria penetrates the fish and then migrates toward the eye lens within hours to, at most, about a day. Once the parasite reaches the lens, it has arrived at what multiple papers describe as an immunologically inert or immune-privileged site. That means the host has only a narrow window in which to stop it before the battle changes shape entirely. For the fish, this is a race. For the parasite, it is a sprint followed by a siege in a protected room. The whole contest is delicate not because it is gentle, but because timing matters more than brute force. If the fish slows the parasite early enough, it may reduce infection. If it fails, the parasite reaches a place where ordinary immune access is much weaker, and the host is left managing damage rather than clearing the intruder cleanly. That early phase is where innate immunity does most of the work. Innate immunity is the fast response, the older defense system, that does not wait to build a highly specific memory before acting. In studies of D, Pseudospathicium in sticklebacks and trout, researchers repeatedly emphasize that the parasite's very short contact time with the host's outer tissues and internal pathways means the initial struggle is driven mainly by innate mechanisms rather than by a slow, polished, adaptive response. The 2014 PLOS gene expression study found that infection triggered innate immune gene activity in sticklebacks and that complement-related responses were especially important. The same study reported upregulation of genes associated with macrophage activation, cytokine signaling, and complement pathways, including C4a, with evidence that different parasite genotypes could elicit different innate response patterns. That matters because it tells us the host is not simply passive while the parasite passes through. The fish does recognize the invasion, and it does answer. The problem is that the answer must come quickly and under pressure, while the parasite is already moving toward a safer anatomical refuge. Older experimental work gives that early response a more physical shape. The abstract of the 1989 study on rainbow trout macrophages reports that diplostomules were capable of eliciting respiratory burst activity from macrophages in vitro, suggesting that reactive oxygen species may play a role in the host's defense. That is the immune system in a very direct mood. Cells are not merely noting that a parasite has arrived, they are launching one of the classic rapid methods for damaging intruders. The older literature cited in later reviews also points to cercaria and diplostomules eliciting immune responses in rainbow trout, and to activated macrophages and specific antibody having some protective role. So there really is combat at the tissue level, and not in a vague metaphorical sense. Fish immune cells can react, can become cytotoxic, and can damage larval flukes. The difficulty lies in the fact that the parasite is built for exactly this phase of danger. It has a short journey to complete, and once that journey is over, the terms of the fight become much less favourable for the host. This is one reason the fish's body condition and baseline immunocompetence matter so much. A 2006 parasitology study comparing lake and river sticklebacks found that fish from a lake population where eye flukes were prevalent were significantly less susceptible than river fish even after a single exposure, which the authors interpreted as evidence that innate immunity plays the major role in defence against this helminth. Those lakefish also showed higher immunocompetence by measures such as respiratory burst activity and spleen size. That is an elegant result because it suggests the battle is not fought equally in every host population. Some populations appear to be better prepared because they have coexisted with the parasite long enough for local adaptation to shape their defences. The parasite attacks quickly, but it does not attack a uniform opponent. In some waters, the fish lineages have been rehearsing this particular emergency for generations. They are not invulnerable. They are simply less easy to surprise. The same idea reappears in later work on sequential infections. The 2014 study on lake and river stickleback ecotypes again emphasised that dusty Pseudospathicium reaches the immunologically inert islands within 24 hours, and the abstract reported differences in susceptibility and immune responses between fish from different habitats. Earlier work found that repeated exposures only marginally decreased infection rates within a period of weeks, and the authors concluded that innate defences seemed more important than acquired immunity for this host-parasite combination. This is an important point, because it goes against the comforting assumption that repeated infection will necessarily teach the host to mount a much stronger later defence. Sometimes it does not, or not enough. With eye flukes, the parasite's dash to the lens compresses the available time for the host to benefit from slower, more specific immunological mechanisms. The fish can become somewhat better prepared, but the improvement may be modest because the parasite's route is designed to escape the conversation almost as soon as it begins. That does not mean adaptive immunity is irrelevant. It means it is handicapped by the schedule. The 2022 rainbow trout study is useful here because it examined immune-relevant gene expression after exposure and re-exposure. The authors note that the Sicari contact the fish surface only very briefly during penetration, and that diplostomules reach the immune-privileged lens within hours to days. They also report that repeated exposure resulted in a slightly lower infection success rate, which they suggest may reflect higher trapping or elimination of diplostomules by humoral and cellular innate elements in primed fish. At the same time, their main finding was more awkward than triumphant. Rainbow trout genetically selected for resistance to a different skin parasite were actually more susceptible to eye fluke invasion and showed a weaker response to secondary eye fluke infection. That result is almost painfully revealing. It suggests that resistance is not one general shield that can be raised equally against every invader. A host may be well defended against one parasite biology and badly matched to another. The battle is delicate in part because the immune system is making trade-offs all the time, and what helps at the skin surface for one enemy may not help much against a larva racing through tissues toward the lens. The parasite, for its part, is not merely surviving the host's defences by luck. It benefits from where and how it moves. Research on host range patterns has suggested that species occupying immunologically privileged sites, such as the lens, may be able to infect a wider and more diverse range of hosts because they face weaker selection for host specificity there. That is a sharp insight. If a parasite hides in a tissue where immune attack is limited, it may not need to match each host's immune peculiarities as precisely as a parasite that remains in more exposed tissues. The lens, therefore, is not just a shelter for the individual fluke. It may also be part of the reason lens-infecting species can be successful across many fish hosts. From the host side, this means the battle is partly lost by anatomy before it even begins. The eye lens is useful to the fish for vision, but it is also a location where immune surveillance is constrained. The parasite has evolved to treat that constraint as an invitation. Even so, hosts are not limited to classic immune chemistry alone. Behaviour itself can become part of the defence. A 2004 parasitology paper examined both acquired resistance and avoidance behaviour in rainbow trout and found that fish reacted to parasite Sicarium by avoiding the infection source, which reduced parasite establishment. The authors concluded that fish may defend themselves more effectively by combining behavioural avoidance with physiological resistance. That is a lovely correction to the image of host defence as something confined inside blood and tissues. The battle begins before penetration, if the host can detect danger in the water and move away from it. In natural settings, this may matter quite a lot. A fish that can avoid a patch of water rich in Sicari is not merely being skittish, it is using behaviour as the first immune organ. Of course, this defence has limits. The same study found that fish held under restricted conditions still became infected and developed cataracts, suggesting that acquired resistance alone was not enough where avoidance was impossible. So the battle has an outer ring and an inner ring. First, avoid the parasite if you can. Second, attack it fast if it gets in. And third, if it reaches the lens anyway, live with the consequences as best you can. What makes the struggle even finer-grained is that it can depend on the identity of the parasite, not just its species. The 2014 PLOS transcriptomics study showed that different parasite genotypes triggered different innate immune gene expression patterns in sticklebacks, particularly in complement genes. The authors interpreted this as evidence that innate immunity can induce redundant but genotype-specific responses. That is a striking result because it means the host is not fighting a generic flatworm in some broad categorical sense. It may be responding differently to one clone than to another. This is where the word delicate earns its keep. The battle is not simply parasite versus host. It is one host genotype with its own immune architecture and history, facing one parasite genotype with its own route, pace, and molecular signatures. In some combinations, the host may trap more larvae. In others, the parasite may pass through more cleanly. Evolutionary biology often sounds abstract until it arrives in a sentence like that. Then it becomes very concrete indeed. A fish and a parasite meet, and the outcome depends partly on who exactly they are. There are hints too that the host can respond not only by killing parasites but by physically managing where they end up. The recent study on bullhead catfish and Diplostomum-induced sac formation suggests that lens-associated sac formation and parasite displacement may represent a host reaction that mitigates the negative effects of infection and impaired vision. Histology showed epithelial proliferation beginning early after infection, and the authors proposed that this process might be a defense or tolerance mechanism. That is a very interesting kind of reply from the host. Instead of fully eliminating the parasite, which may be impossible once it has reached the eye, the fish may alter tissue organisation in a way that pushes the parasite away from its most harmful position. This is not victory in the clean sense, it is damage control. Yet damage control is one of the main ways real organisms survive. The host does not always need to erase the enemy. Sometimes it needs only to reduce how much of the eye the enemy can ruin. The parasite's advantage, however, remains severe. It is moving through a short immune-exposed corridor toward a much safer compartment, and the host's defences are strongest exactly where time is shortest. That asymmetry explains why the struggle can feel almost unfair. A lake-adapted stickleback may be less susceptible. A rainbow trout may show early upregulation of cytokines, cathelicidins, interferon gamma, interleukins, and other immune genes after exposure. Macrophages may produce respiratory burst activity. Complement cascades may be activated. Re-exposed fish may trap a few more larvae than naive ones. Behavioural avoidance may reduce contact in the first place. All of that is real. None of it changes the central difficulty. The parasite does not need to survive in open conflict for very long. It needs only to get through the door before the host can close it completely. This is why studies of local adaptation and immunogenetics are so central to the eye fluke story. They show that the host is not helpless, but also not sovereign. Lake sticklebacks in symmetry with eye flukes can be less susceptible than naive river populations. Innate immune responses can vary with parasite genotype. Host genotype can shape susceptibility. Yet previous exposures may yield only marginal gains, and resistance to one pathogen may even correlate with greater susceptibility to another. That is not a simple arms race in which one side merely becomes stronger and wins. It is more like a constant adjustment of narrow margins. The host improves a little here, the parasite slips past a little there, the habitat changes the odds, and the contest goes on. From the outside, one sees a fish with cataracts or a parasite in a lens. From the inside, what has happened is a sequence of very small successes and failures stacked tightly together over a few critical hours. And that, perhaps, is the most unsettling part of the whole interaction. The battle is delicate because both sides are highly specialised, and neither is acting in broad dramatic gestures. The fish is mounting oxidative bursts, complement activation, cellular recruitment, altered gene expression, and sometimes avoidance behaviour. The parasite is racing toward a tissue the host cannot easily police, exploiting anatomy as much as physiology. Sometimes the fish holds it back. Sometimes the parasite reaches the lens. Sometimes the host's answer becomes tolerance rather than elimination. The result is not a clean story of resistance or defeat. It is a fine, ongoing negotiation between invasion and control, between damage and compensation, between what the host can do quickly enough and what the parasite can avoid long enough. And once that struggle is understood at the level of bodies and tissues, it becomes easier to see why eye flukes matter beyond individual fish, because the same small contest, repeated across many hosts in the same water, starts to tell a much larger story about food webs, environmental conditions, and the way hidden parasites can quietly shape entire aquatic worlds. What eye flukes reveal about oceans, ecosystems, and survival is larger than the parasite itself, and larger than the eye it occupies. Once you stop looking at the fluke as a single unpleasant detail inside a single fish, it begins to show you how aquatic life is actually organised. not as a neat list of separate creatures minding their own business, but as a network of dependencies, accidents, pressures, shortcuts, and consequences. That is one of the reasons parasitologists and ecologists take parasites seriously, even when everyone else is trying very hard not to think about them during lunch. Parasites with complex life cycles do something unusually informative. They tie multiple hosts together in a way that makes them useful indicators of food web structure, biodiversity, and environmental stress. Marco Leese's widely cited review put the point plainly. Parasite populations and communities can reflect trophic position, the presence of other organisms needed to complete the life cycle, and changes in ecosystem structure and function. In that sense, a parasite is not just a disease agent, it is also a piece of ecological evidence. Eye flukes are particularly good at revealing that because their route is so demanding. A trematode with a snail host, a fish host, and a bird host cannot complete its life cycle unless all of those players are present in the same time and place, and unless the water body still permits their interactions to happen often enough. That is why parasitologists sometimes describe complex life cycle parasites as indicators of species diversity. If the parasite is present, then its hosts must also be present, and if any one of those hosts drops out of the system, the parasite loses its route and dies with it. A 2012 study comparing parasites and insects as indicators in freshwater lakes emphasized exactly this. Trematodes with complex life cycles can be ideal indicators of species diversity because they require multiple hosts, and if one of those hosts is absent, the life cycle fails. Eye flukes therefore tell you that a lake or estuary is not biologically empty. It still contains enough snail habitat, enough fish exposure, and enough bird predation to keep a complicated cycle alive. That is not a cheerful message, perhaps, but it is an informative one. That leads to one of the more counterintuitive ideas in parasite ecology, which is that a healthy ecosystem is not necessarily a parasite-free ecosystem. In fact, some ecologists argue almost the opposite. Marcogliese wrote that parasites are natural parts of all ecosystems, and that healthy ecosystems have healthy parasites. That line tends to surprise people because the ordinary human reaction is to treat the word healthy as the opposite of parasitized. But ecosystems are not judged the same way we judge a sore throat. A diverse parasite community can indicate a diverse and functioning host community, a complete food web, and enough ecological continuity for long life cycles to persist. If a system is simplified, overfished, polluted in a certain way, or stripped of key hosts, parasite diversity may fall, not because the ecosystem has improved, but because it has become too damaged or too incomplete to support its former complexity. In other words, the absence of parasites can be a sign of cleanliness in one narrow sense, and collapse in a broader one. Nature is often rude enough to make both things true at once. That does not mean all increases in parasitism are good news. Here the eye fluke becomes especially revealing, because it shows the difference between parasite diversity as a sign of ecological complexity, and parasite outbreaks as a sign that certain environmental conditions are favouring transmission too strongly. Lafferty's review on ecosystem consequences of fish parasites notes that eutrophication and thermal effluent often raise rates of parasitism in aquatic systems because increased productivity can boost intermediate host abundance. For trematodes, and eye flukes are trematodes, that often means more snails. USGS researchers working in Upper Klamath Lake have been making exactly that connection in practice. They note that snails can shed tens of thousands of infective parasites into the water each day. And another USGS summary explains that highly nutrient-rich waters could mean a greater abundance of snails, which in turn means more eye flukes. So the parasite does not simply tell you that a food web exists. It can also tell you when the balance of that food web has shifted in a way that makes one stage of the cycle too successful. That is part of why eye flukes matter so much in conservation contexts. In Upper Klamath Lake, researchers are not studying them out of idle curiosity. or because someone in a lab developed an unfortunate fondness for cloudy fish eyes. They are studying them because endangered juvenile suckers are living in a system where birds, snails and fish all overlap, and where eye flukes may be contributing to mortality by blinding fish, damaging movement, and making them easier prey. USGS materials describe the lake as a haven for trematodes, because it contains large colonies of birds and an abundance of snails and fish, and they note that infected fish are often blinded and then eaten by water birds, completing the cycle. Once you see the problem at that scale, the eye fluke stops being an oddity and starts being an ecological pressure with management consequences. It can become one piece of a larger story about juvenile recruitment failure, water quality, habitat alteration, and the survival of fish populations that are already under stress. Eye flukes also reveal that survival in aquatic systems is often decided by very small handicaps rather than dramatic catastrophes. A fish does not need to be killed immediately by a parasite for the parasite to matter. It only needs to become slightly worse at feeding, slightly slower to detect danger, slightly less coordinated in a school, slightly more likely to make the wrong movement when a bird passes overhead. That is why ecologists are so interested in parasite-induced changes to host behaviour. The scientific report study on juvenile European perch showed that increasing eye fluke infection intensity altered diet composition, making heavily infected fish more selective and changing how they interacted with lower trophic levels. The authors went further and suggested that eye flukes may play an important role, not only in energy transfer to upper trophic levels, by increasing susceptibility to predation, but also in host interactions with lower trophic levels by altering diet composition with possible cascading effects on zooplankton and phytoplankton. That is a very large consequence for a very small flatworm. This is where the eye fluke becomes more than a parasite story and starts to look like a food web story. Parasites are embedded in food webs because they depend on trophic transmission, and Lafferty's review makes the case that without parasites, no food web effectively represents the trophic dynamics of a system. That sentence is worth appreciating for a moment because it quietly overturns the usual picture of ecology. We often draw food webs as though predation, competition, and primary productivity are the whole drama, and parasites are an unpleasant footnote, but parasites add links, redirect energy, alter host behaviour, affect who eats whom, and in some systems account for a meaningful amount of biomass. In estuarine work reviewed by Lafferty, parasites altered important network statistics such as connectance and nestedness, and the broader implication was that leaving parasites out gives you an incomplete map of the ecosystem. Eye flukes fit that logic very well. They connect snails to fish, fish to birds, and host behaviour to predation risk, all while changing feeding at lower trophic levels. They are not outside the web, they are one of its smaller but very active threads. They also reveal something important about habitat. Eye flukes do not favour just any water. They favour water with overlap, slow or sheltered enough for snails, busy enough for fish, open enough for fish-eating birds, stable enough, at least for part of the year, for all three to keep meeting. That is why the same basic ecological logic appears across ponds, lakes, marshes, reservoirs and brackish coasts. The classic diplostomum systems are often fresh water, but the pattern is not confined there. In the Baltic region, round gobies in Kalmar Sound were found with an eye fluke prevalence of 90 to 100%, and the study reported that heavier cataracts were associated with weaker responses to simulated avian attack. In southeastern Baltic sea cod, diplostomum eye flukes were also recorded, with a prevalence of 7.4% and a mean intensity of 11.9 parasites per fish in one 2013 to 2014 survey. So when we use the word ocean in a topic like this, the most accurate lesson is not that classic eye flukes are ocean exclusive, because many are not, but that the same ecological design extends outward toward coastal and brackish systems where sea, river and shoreline life begin to mix. That brackish and coastal extension matters because it reminds us not to think in rigid habitat boxes. A parasite does not care whether humans label a place as lake, estuary, lagoon, bay, or marginal sea so long as the route still works. If snails release infective stages, fish acquire them, and birds consume the fish, the cycle can continue. The fish species may change, the salinity may shift, and the local bird assemblage may differ, but the ecological requirement remains the same: connected hosts. This is another reason parasites are informative. They reveal connectedness better than many more charismatic organisms do, because they physically fail when that connectedness breaks down. A bird can still fly over a damaged wetland. A fish can still survive for a while in a simplified estuary. A parasite that needs both of them, plus the right snail, is a much stricter judge of whether the system is truly functioning as a linked whole. Flukes also reveal how environmental change can reshape survival indirectly, rather than only through obvious toxic effects. Lafferty notes that toxic pollutants are generally associated with lower parasite abundance in many fish systems, while eutrophication can raise parasitism by favouring intermediate hosts. That means parasite patterns can move in opposite directions depending on what kind of disturbance is acting on the system. A heavily contaminated site may lose parasite diversity because free-living stages and intermediate hosts are impaired. A nutrient-enriched site may gain transmission pressure for generalist trematodes because snails or other intermediate hosts become more abundant. This is one reason parasite data are useful to managers. They do not tell one simple moral story in which more parasites always means a worse ecosystem, or fewer parasites always means a better one. Instead, they help identify what kind of environmental process may be dominating. With flukes in particular, high transmission can be a clue that host overlap, snail abundance, and habitat conditions are favouring the life cycle in ways that matter for fish survival. There is another, more uncomfortable lesson in all this, and it concerns what survival actually means in nature. We often think of survival as a direct contest between predator and prey, strength and weakness, hunger and escape. Flukes reveal that survival can also hinge on a hidden third party, changing the quality of perception itself. The fish thinks it is in a contest with prey below and birds above. In reality, the parasite is already inside the contest, degrading the fish's ability to read it. This is not merely a medical problem happening inside one body. It is a reallocation of odds within the ecosystem. A more heavily infected perch may feed differently. A heavily cataracted goby may respond less effectively to avian danger. A blinded juvenile sucker may become easier prey. Each of those is a small revision to the probability of one organism surviving another hour. Stack enough such revisions together across a season, and you begin to alter which fish grow, which birds feed, which prey populations are suppressed, and which cohorts fail to recruit into adulthood. That is ecosystem change happening through perception. They reveal too that biodiversity includes things we do not like. There is always a temptation to speak of conservation as though it means preserving only the creatures we admire or the ones that photograph well. Parasites resist that sentiment very effectively. They are part of biodiversity whether we welcome them or not, and they are often deeply tied to the structure of the communities they inhabit. Mark Elise's review argues that parasites can provide important information on environmental stress, food web structure and function, and biodiversity. Lafferty likewise notes that complex ecological networks can support richer parasite communities. Taken together, those ideas lead to an awkward but useful conclusion. If you remove hosts, simplify habitats, truncate food chains, or otherwise flatten ecosystems, you do not just lose birds, fish, and invertebrates. You also lose the parasite relationships that depended on them. No one will usually put that on a postcard, but it is still true. A damaged ecosystem is often one that has lost not only beauty and abundance, but also much of its hidden biological complexity. And yet flukes also remind us that not every complex system is a comfortable one. Complexity can be intact and still brutal. A lake full of birds, snails and fish may be ecologically rich while also being a very bad place to be a juvenile fish with an infected lens. That is perhaps the most honest lesson parasites offer, Nature is not arranged around fairness, it is arranged around continuation. Flukes continue by moving through intact host networks, by taking advantage of productive shallow habitats, and by converting small visual handicaps into transmission opportunities. The fish continues, if it can, by feeding well enough, hiding well enough, and avoiding being the individual whose cloudy eyes finally cost it one mistake too many. The ecosystem continues by absorbing all of these tiny transactions at once, from snail shedding to bird feeding to shifts in prey choice and water clarity. None of it is clean. All of it is connected. So what flukes reveal in the end is not just that parasites are strange. It is that aquatic life is full of hidden negotiations we miss when we look only at the largest animals or the most visible events. A parasite in an eye can tell you that a food web is intact, that a shoreline is productive, that snails are abundant, that birds are feeding, that a lake or brackish bay has the right structure for a 3-host cycle to persist. It can tell you that environmental change is favouring one host stage over another. It can tell you that host behaviour is being nudged in ways that may alter energy flow through the system. It can even tell you that the difference between survival and predation is sometimes no bigger than the loss of visual clarity inside one lens. Once you see all that, the fluke no longer looks like a biological curiosity with unfortunate branding. It looks like a small but very precise witness to how ecosystems really work, and why life under the water, whether in inland lakes or at the edge of the sea, is held together by connections that are easy to ignore right up until one tiny flatworm makes them impossible not to notice. And so, dear listener, we arrive at the quiet edge of this strange little journey, having followed a creature so small that most of us would never notice it, and yet so effective that it can alter the life of a fish, shape a meal for a bird, and tell scientists something important about the health of an entire body of water. We began with what sounded like a simple, slightly alarming subject, the fluke, a parasite that lives in or around the eyes of fish. But somewhere along the way, the story widened. It stopped being only about something unpleasant in the lens of a fish and became something more revealing than that. It became a story about connection, about how one tiny organism can move through different bodies, different habitats, and different stages of life without ever needing to be large, loud, or dramatic. It only needed to be precisely adapted, consistently timed, and patient enough to let the rest of the ecosystem do much of the work for it. That is one of the things I find most striking about the natural world when we slow down enough to look properly. The creatures that change the shape of a system are not always the ones we notice first. Not always the shark, the eagle, the whale, or the bear. Sometimes it's something small and almost offensively easy to overlook. A flatworm, a larva, a drifting stage in the water, a body that spends part of its life in a snail, part in a fish, and part in a bird, without any of those hosts knowing or caring what role they have been assigned in its future. There is a certain blunt practicality to that arrangement. No sentiment, no ornament, no moral lesson built into it. Just one living thing using the habits of several others to continue existing. And perhaps that is what makes the subject linger in the mind. The fluke is not frightening because it is huge or fast or obviously predatory. It is unsettling because it is organised. It has no need to rush, no need to terrify, and no need to dominate the entire world around it. It only needs to complete its route. That is enough. A bird feeds, a snail sheds larvae, a fish enters the wrong stretch of water at the wrong moment, or rather the right moment, if one is speaking from the parasite's point of view. And suddenly, something invisible has begun to shape what that fish can see, how well it can feed, how quickly it can react, and how long it may remain part of the living world below the surface, instead of becoming part of the digestive arrangements of the world above it. There is something very humbling in that. We humans are rather fond of scale. We like the big animals, the dramatic weather, the towering trees, the obvious forces. We are drawn to what announces itself. But much of life is governed by things that do not announce themselves at all. Hidden larvae, chemical signals, immune responses, shifts in light, changes in water temperature, the abundance of snails along a shallow margin, the angle of a bird's descent, the transparency of a lens inside a fish no one will ever name. An entire chain of events can hinge on details so small that if you took a casual walk along the bank of the lake, you would never suspect any of it was happening. The water would seem calm, the birds ordinary, the fish invisible beneath the glare. And yet inside that calm scene, bodies would already be negotiating survival in ways far more complicated than the surface would ever suggest. That, I think, is one of the most useful lessons hidden inside a topic like this. The world is never only what it looks like from a distance. A lake is not just water with fish in it. A coastline is not just waves and birds and reeds. Every place that appears simple from far away becomes crowded once you look closely enough. Crowded with lives, with strategies, with tiny contests and quiet dependencies. The fish is not only avoiding predators and seeking food, the fish may also be carrying a larval parasite in its lens. The bird is not only feeding, it may be serving as the final host in a cycle that has repeated itself for generations. The snail on the stem is not merely part of the scenery. It may be the place where one successful invasion becomes thousands of new attempts. Once you know this, the scene does not become less beautiful. It becomes more complete. A little less innocent, perhaps, but more complete. And there is something strangely fitting about all this taking place in an eye. Not the stomach, not the liver, not some hidden tissue that most of us rarely think about. The eye. The part of the fish that turns light into information, motion into warning, shape into opportunity. The organ that lets the animal read the water around it and react with the speed its life requires. If you wanted to choose one place where a small amount of damage could have consequences far beyond the size of the injury itself, the eye would be a very effective choice. A clouded lens is not simply a local defect. It is a change in how the world arrives. A damaged retina is not only tissue damage, it is a change in what the fish can trust, what it can track, what it can judge in time. Underwater, where light is already altered by depth, particles, reflections and movement, that loss of clarity matters very quickly. And yet even there, the world remains practical rather than theatrical. The fish does not pause for a tragic monologue. The parasite does not twirl a tiny moustache in satisfaction. Life continues in the plain, stubborn way life usually does. The fish still swims. It still feeds if it can. It still tries to detect danger. It still moves through the same water with whatever ability remains to it. That is another thing nature does without apology. It keeps going while conditions worsen. It does not stop the story to point out that something unfair has occurred. It simply lets the next moments happen. Some fish cope longer than others. Some avoid the heaviest burdens. Some are caught sooner. Some make it through. The parasite continues where it can. The birds feed where they can. The snails go on being snails with all the calm, personal ambition one expects from a snail. And the lake, for all its hidden transactions, still looks like a lake. I suspect that is why people are often so drawn to the hidden side of biology, even when it is uncomfortable. There is honesty there. A parasite does not flatter our ideas about nature. It does not pretend the world is tidy or fair or arranged for our emotional comfort. But it does show us that living systems are deeply connected, and that those connections are often much more specific than we assume. The eye fluke's life only works because birds, snails, fish, water, and timing all line up often enough. Remove one piece and the root breaks. Increase one piece too much and the root may become more intense than before. Change the habitat and you change the parasite. Change the parasite and you may change the behaviour of the fish. Change the behaviour of the fish and you may alter the odds of predation, feeding, and growth. It is all tied together, and often by pathways so small that we would miss them completely without science. Science, for all its instruments and jargon and habit of giving things wonderfully unromantic names, is often at its most beautiful when it notices what everyone else has missed. Someone looked at a cloudy fisheye and didn't stop at saying, "Well, that seems bad." Someone asked what was inside it. Someone linked the fish to the snail and the snail to the bird and the bird back to the water. Someone measured cataracts, traced larvae, checked retinal damage, tested escape responses, and sat long enough with a deeply inconvenient little flatworm to understand that it was not merely causing a local problem. It was revealing a pattern. And because of that patient work, what would otherwise remain a hidden injury in a hidden animal becomes something we can understand as part of the structure of life itself. There is comfort in that, oddly enough. Not comfort in the parasite. The parasite remains thoroughly committed to being unpleasant. But comfort in the fact that the world can be known more deeply than first appearances suggest. Comfort in the idea that even the things that seem obscure, minor, or grotesque still belong to an intelligible system. If you watch carefully enough, if you compare, test, observe, and return again and again to the same small puzzle, patterns begin to emerge. And once patterns emerge, so does a different kind of calm. Not the calm of believing everything is harmless, but the calmer and better thing, which is understanding how it fits. Perhaps that is what this channel has always been about in its own quiet way. Life beyond seen. Looking a little longer at what slips past casual attention, giving space to the parts of science that do not need to shout in order to be remarkable. Sometimes that means stars and time and strange questions about the universe. Sometimes it means a flatworm in the eye of a fish. Both are, in their own ways, reminders that the world is larger than whatever we happen to notice first. And if that feels a touch humbling, that may not be a bad thing. Humility is often just curiosity that has learned some manners. If you're still with me now, perhaps a little sleepier than when we began, then we have at least done this small journey properly. We have taken something odd and made it familiar, then taken something familiar, the calm surface of water, the ordinary eye of a fish, the simple act of a bird feeding, and made it strange again. That feels like a fair exchange. The hidden world deserves both responses. Familiarity enough that it can be understood, and strangeness enough that it can still be respected. And now, having spent this time with such a small and secretive creature, it may be worth returning to the simplest image of all. One eye, just one. Not a vast ecosystem, not a map of food web interactions, not a long Latin name in a paper, but one small fish eye in a body of water at dusk, clear or clouded, healthy or burdened, watching the movement of the world as best it can. There is something very direct about reducing the whole story to that. Because in the end, all the complexity we have talked about today, all the snails and birds and larvae and tissues and habitats, becomes real in one place at a time. One fish, one lens, one life moving through water with more or less clarity than before. That image lingers with me because it reminds me how much can be contained inside something small. A single eye can hold damage, adaptation, vulnerability, biology, ecology, and the outcome of an evolutionary strategy millions of years in the making. It can also hold, in a very literal sense, the difference between seeing danger in time and not seeing it at all. We tend to think important things should arrive with scale, but sometimes importance arrives as a blur. Sometimes it arrives as reduced contrast, slower response, a missed prey item, a delayed turn. Sometimes it arrives in tiny fractions that add up to fate. And if that sounds a little severe, well, nature often is. But it is also full of persistence. The fish persists if it can. The host fights where it can. The ecosystem persists by carrying all these relationships at once, including the ones we find distasteful. Life does not wait for our approval before becoming complicated. It simply goes on being complicated. And perhaps the best we can do, as observers, is to notice honestly and describe carefully, not to force the world into a prettier shape than it has, but to see it clearly enough that even the least charming creature has its place in the story. So, as the evening settles in a little further, or the morning light begins somewhere on the other side of the world, let me ask you once more, gently, in the spirit of all these hidden connections, where are you listening from right now, and what kind of water comes to mind in your corner of the world? A harbour, perhaps, a quiet pond, a broad river, a cold estuary, a lake with reeds along the edge and birds lifting off at first light. Whatever water you picture, I hope this strange little journey has made it feel a bit fuller, a bit more detailed, and perhaps just a little less empty than it did before. Not because there is something to fear in every ripple, but because there is more life in every place than we usually allow ourselves to notice. And if you found something soothing in that slower way of looking, if this quiet detour through parasites, fish and hidden ecology gave your mind something gentle to rest on, then do leave a like, perhaps subscribe, and share this sleepy corner of science with someone else who enjoys learning about the natural world at a calmer pace. It helps this little space continue, and it helps these curious, unlikely stories find the people who might enjoy them most. For now, though, there is no need to think any harder about lenses, larvae, or the private complications of snails. You have done enough careful looking for one night. Let the water settle. Let the birds drift out of sight. Let the fish return to the dim green world below. And let that single eye, strange as its story has been, become simply one more reminder that life is always busier, smaller, and more intricate than it first appears. Sleep well, wherever you are, and whenever this finds you.